It's now 11 o'clock, so we'll get started. And I want to thank you for taking the time to come to this meeting and make sure that all the staff members that you want on this meeting are here at this meeting. I'm just going to go through a little introduction of things that we've done in the past and hopefully where we can go in the future. And hopefully my screen is sharing. On my presentation, we have a timeline in which we as a community have been talking about biomarkers. In January of 2018, we required the biomarker core as a component of the ADRC. And this was done in RFA 19001. And we instructed that the biomarker core should collect, store, track, share, and analyze biomarkers. They could be standard or novel, and they should contribute to the understanding of the heterogeneity, the onset or progression of disease symptoms, or improved diagnosis. And the underlying portion is kind of like where we're at right now. Catalogs of available biomarkers and relevant data should be shared with NAC, and samples may be shared through NICRAD. And we still have to kind of work through the logistics of making that happen. In April of 2018, knowing that we have biomarkers being required, we had a biomarker meeting in Los Angeles in which we launched an effort to standardize CSF biomarkers with mixed results. ADNI shared their Roche data with us. Kai Blenau traveled all the way to Los Angeles to discuss neurofilament light chain protein. Mark VCID shared best practices. We discussed in 2018 opportunities for a center network, biomarker initiative, and digital biomarkers. So the conversations 3 years later are pretty similar. In May of 2019, we worked with our colleague, Austin Yang, to have a translational biomarker session at the meeting in Philadelphia. And we brought in a colleague from FDA to discuss AD diagnostic tests and the regulations of them. We shared advances in clinical mass spectrometry and gave an introduction to TPAD 2.0, which is a project entitled The Next Generation of Translational Proteomics of AD. And the AMP-AD biomarker discovery pilot projects was shared to you. And then in May of 21, which was just a few months ago, we had another kind of biomarker core meeting where NICRAD updated everyone on the biomarker core lab plans that they have at NICRAD. Mark's VCID came back to join us again and present their progress. And I presented a review of our biomarker inventory, what items we already have in stock to hopefully begin leveraging the center network with the resources that we have. And the next slides that I have to show are going to repeat this inventory. And so the little pink X here on the timeline is the time in which all centers currently now have a required biomarker core. We're going through the competing process between the years of 2018 and 2021. So now all 33 centers have a required biomarker core and should be following the instructions of the RFA to have catalogs of available biomarkers, and the relevant data should be shared with NAC and samples shared through NICRAD. So then that brings us to today's meeting, where we had a call for volunteers to present on biomarker topics. So this agenda was totally homegrown, volunteer-based. And it's our mid-year check-in in 2021. before establishing the NAC will establish a biomarker core steering committee, which I've kind of been talking about for like a whole year. And in this biomarkers core steering committee, which the election will happen in January of 22, we will elect 7 members in a laddering term of service to establish the membership of the core of the steering committee. And NAC will put out a call in the listservs for nominees and conduct the election. That hopefully everything will be in place for the May spring meeting. So that's kind of where we're at in the creation of the biomarker core. And I'm hoping that today's meeting provides all of the background information that whoever volunteers to be on the biomarker core steering committee can start with this flesh background.
Maybe we could go to the next speaker while we wait for her to reconnect.
Sure, we could do that. And so our keynote presentation is Dr. Tansey, if you would like to pull up your slides.
Sure.
Yes, you can get started. Thanks for rearranging.
Yeah, no worries. So yeah, thanks for inviting me to talk to this amazing group. I'm not sure who I owe the payback to, but I have a pretty good feeling I know. When I was putting this together, I had been talking to my colleague, former Emory colleague, William Hu, and we've had a lot of conversations about this, and he and I worked together when we were both at Emory. So what you're going to hear is kind of a combination of both of our thoughts about this, and you know, I'm probably going to give you the punchline in that, you know, you're probably not going to hear what you want to hear, but here it goes. So first of all, you all know that there's plenty of evidence in Alzheimer's that biofluids have inflammatory markers, and you're looking to leverage what has been collected. And, you know, why do we need a biomarker? Used to be that we wanted to distinguish it from, you know, a disease case from a control. And we could also be using it to stage, to look at endophenotypes, predict trajectories, aid in clinical trial design and monitoring, and obviously predict responsiveness. I think, as you'll hear, the time to distinguish case versus control is now gone. I think what we're trying to figure out now is how to look at them for endophenotyping and stratifying and looking at the heterogeneity in these diseases. I think the thing that's really difficult about biomarkers, and I'm outlined here in red, is that an ideal biomarker has a lot of features that unfortunately inflammatory biomarkers do not give us. And those are outlined here, right? So easy to measure, you know, a lot of them are below the lower limit of detection in a lot of these assays. Reliable and consistent, robust, high abundance, does not change throughout a 24-hour period. I'll give you examples of why they do. Long life, specific to disease condition, most of them are not. And then minimally influenced by environmental or external factors. And when we know that that is definitely not the case, we have a lot of these factors being influenced by stress and sleep and you know, diet and medications. And this is a schematic from William, where Alzheimer's biomarker publications in both CSF and blood are increasing, right? Have increased markedly over the past few decades. And so this is reflecting both obviously interest and our ability to measure and certainly look for things that may be of interest. And so the question at hand that Cerise asked me to sort of, think about and address with you is there a way to leverage data specimens that the ADRCs have to begin framing an assessment, the ability to perform comprehensive assessment of longitudinal, immunological, and AD biomarker information in richly phenotyped, demographically diverse cohorts? And she told me that this is what you guys have. So, plasma and serum is what I call, what's in the pipeline. And I hope to convince you that, while that is useful and easy to get, many of us have been finding out that, you get not a lot of information about the inflammation trait of the patient. You get information about the inflammation state, because it's a snapshot in time. And that if you want to get information about the inflammation trait or innate immune dysfunction or adaptive immune dysfunction, that you may need to get the PBMCs and plate them and challenge them with an immune challenge, and then look and see, are they hypersecreting or hyposecreting or, not phagocytosing well. And so that may be more informative, clearly more difficult to do, more expensive, etc. But, I think that may be something that you may need to consider in the future. So I like to use the schematic from Tony. I had no idea he was going to be here, but I use it in most of my talks to just remind you that, the presence of cytokines and chemokines is something that is normal and important, because these are part of the bidirectional communication between all our tissues, and so we have to have them. And so, only when they're in excess or out of balance do we have big issues. And so because of the increased longevity and inflammaging and all this, that things are out of whack, and because of the immune aging, we have this age-related immunodeficiency or age-related inflammatory syndrome, and impaired adaptive immune responses constitute of low-grade inflammation, and you have a fair amount of immune dysfunction, if you will, as you're aging. And so is there any evidence that this is in fact the case, and what are some of the factors that support that idea? And so one of the things that immune cells produce as they are activated, and one of the usual suspects and culprits that we and others have been studying is TNF. And so I'm going to use the TNF as an example of something that we've been tracking. And interestingly enough, William has pulled out the soluble TNF receptors as markers as well. And so TNF comes in two different flavors. It is important for inflammation, immunity to infection, chronic disease, but it comes in two different forms. Lymphoid organ development is sustained by the membrane-bound form, and then it's cleaved to a circulating form, and then when it's cleaved by TACE, it's a soluble TNF ligand that is sustained, mostly physiologically paired with TNFR1. And so we know that TNF levels increase early in life and correlate with aging, and TNF levels, as well as TNF receptor 1 and TNF receptor 2, the soluble forms, along with IL-6, are linearly and positively associated with age. And so this is interesting because this appears to start going up around age 30 when things start to go in reverse. And the evidence for, soluble TNF increases, there's many studies, but including some of the ones here, is that, Howard Phillott, in fact, was one of the first that reported soluble TNF increases in plasma in AD subjects. Cerebrospinal fluid has increased TNF. Plaques have co-localized TNF with it. And then there's several studies, not all, that show some increases with disease progression. And so what does it matter? And does it matter that you eliminate some of these TNF cytokines or others? And so not direct evidence and not to imply causation, but some, medical claims and epidemiological studies suggest that elimination of TNF may in fact give you something, and that's the evidence that people with autoimmune diseases may have increased incidence of dementia unless they are on anti-TNF blockbuster drugs, and then that incidence drops, precipitously. And the similar evidence that anti-TNF drugs in people with autoimmune diseases also lowers the incidence. And so, it's something that makes us take notice and ask the question, does, the inflammatory milieu of the body have an effect, and should we be paying attention to TNF or all others? So the chronic inflammation precedes and drives development of dementia, and starts early, and the dysfunction is maybe associated with cognitive decline. And is it good or bad? And so we think that that things may be similar to the way the cancer field is thinking about it, that an acute inflammatory trigger is adaptive and short duration and leads to classic myeloid cell activation, but a chronic long duration activation signal may lead to pathological myeloid cell activation. And so I think about something very similar for inflammaging and metabolic syndrome and chronic systemic disease, that it's very similar to what you would have in a cancer or chronic infection syndrome, that perhaps those chronic systemic diseases are leading to weak activation signals and mild but consistent elevation of innate immune cell activation, both systemically and centrally, and pathologic myeloid cell activation that's leading to neuroinflammation and neurodegeneration. So I think we can take some ideas from them and approach some of the techniques that they have been using to think about it in certain ways. So in terms of other transmitters, what do we need to think about? So gliotransmitters come into play. A lot of things that astrocytes and glia make are added to the mix. TNF is a gliotransmitter. A lot of things are, produced that make things a little bit more complicated because they are not just inflammatory factors, they're also serving communication roles, like in Tony's schematic. And then the other complicating thing is that not only do these inflammatory molecules activate these cells, but they're made by these cells. So they're both upstream and downstream of the cells that are making them and that are being made. And so one question to ask is, what biological processes do the markers reflect? And so, William likes to separate these two into what people like to use, the hypothesis-based approach to finding biomarkers versus a discovery-based approach. And the discovery-based approach is usually the more common one. But the hypothesis-based approach is, brain biomarker relationships should be established in controls, wild type, and separately in cases or disease models. Right, and functional assays of cells isolated from fresh postmortem brain, and you get the result. In a discovery-based approach, basically you're just looking for a signal, but the problem is that a lot of analytes are below the level of detection of most conventional assays. And the other thing that happens is that you can't really infer a mechanism from something that's high. And so there's usually insufficient evidence to suggest a therapy, right? And so I'll show you some additional examples. And, as an example, do we think that we should be treating everybody with anti-TNF antibodies? Probably not. It's just suggesting that that may be an analyte that, is playing a role in some of the progression. So in terms of detection platforms, one other thing you need to think about, and the reason it's going to be difficult for you guys to harmonize, is that the platforms influence the outcomes, right? And when we were at Emory, we did a bake-off, what we call a bake-off of, you know, different matrices and different platforms. And so whether you measure with Quanterix or MSD or Luminex or Quantikine, you know, different ones, you get different outcomes, right? And so the absolutes may not matter, but sometimes, you know, your ability to detect these in certain populations will matter. And if you have to eliminate analytes because it's not detected in all of your cohorts, then you're going to start to lose power. And so this becomes a real issue and a real problem. The other thing is, you know, some of these perform a little different in CSF versus plasma versus serum. And so a lot of that has to be sort of, you know, initially empirically determined. And also, you know, I think most of us think that for off-the-shelf assays, you know, MSD is great and Luminex is great. And if you're going to do more discovery type things, then maybe Quanterix is your platform. But, you know, we can go into that later. Okay, so the one take-home message that you probably ought to remember, and this is the world round, is that CSF and plasma inflammatory proteins don't correlate well at all in any cohorts that you look at. We've looked at them. William has looked at them. The brain is doing its own thing, especially with inflammatory proteins. They have their own rhythms. We've measured them and published them in Parkinson's patients and others, and there are inherent rhythms, if you will, for cytokines and chemokines, and you know, I know Tony's published book chapters on this that are not seen systemically, right? And so the systemic circulation is not going to reflect what's happening in the brain. The only analyte that we've ever seen correlate well with the central compartment is CRP, and the magnitude is different. So you are an order of magnitude higher in the periphery than in the brain, but it is a perfect correlation, which is really interesting in almost all subjects, wild type controls and cases that we've ever seen. So that's an interesting thing that we can talk about later. But other than that, there's no real correlation. And so this is an example from the Parkinson's study that I mentioned. It was called a 24-hour qualification study, where we looked at 12 PD subjects and 8 controls, sampled over 24 hours, every 2 hours, with a CSF catheter, lumbar catheter, and a peripheral catheter. And we were looking for a whole bunch of different things. What's the best biomarker for distinguishing PD versus control, and you know, could we assign membership to PD versus control with TNF versus synuclein versus what? And so we got some interesting things. Just to give you an idea, the blue ones on the left are those that were variable across time but not different between PD and controls. The ones in the middle are variable across time and different between PD and controls. And the ones on the far right were different between PD and controls and little variability across time. So if you're gonna talk about the ideal biomarker, potentially you could stick with the serum TNF and GAL, which is lipocalin-2, and serum interferon gamma, right? Those would be your potential ideal biomarkers because they're systemic, they're not varying across time, and they're different between PD and controls. But as I said, you know, you can also see that the sampling methodology influenced the outcome, because you see this ratcheting up over time from time 0 to time 24 of CSF synuclein, of A beta, right? So almost like the stress of sitting there for 24 hours kind of increased your outcome, right? If you had actually stuck these people every 2 hours instead of having them have this catheter, maybe you would have gotten something completely different. So the methodologies with which you sample is also going to have, you know, an effect. We had diurnal rhythms that we saw in healthy controls that seemed to be disrupted in PD subjects. And these included TNF, CRP, IL-6, IL-8, and even A beta peptides. And then, as I mentioned before, only the serum and CSF CRP levels were positively correlated at baseline in healthy controls and PD. Everything else, as you can see here, were in completely different directions for healthy controls and PD. And so the central compartment just does not reflect what's going on peripherally. So then this is your typical biomarker analytical approaches and pitfalls. This is from William. General approaches in studying inflammatory alterations, characterizing cellular products, whether it's a secreted or surface shed, whether you're looking at cells or gene expression or ex vivo responses to challenge. You have lots of proteins that have been implicated, obviously from primary cell-based work. And your typical analytical approach is, to run t-tests or u-tests, and you classify, AD versus normal controls. And the pitfalls are, that you have to deal with, the detection, potential, platform differences, and then you don't get much between the control and the case, right? And so this is, I think, where we get stuck, that you don't have the ability to discriminate between the endophenotypes, because nobody really takes it beyond that. And I think anybody could probably tell you the difference between a case and a control, and you don't need inflammatory factors for that much. Principal component and factor analysis. So an alternate framework for optimizing the two approaches would be a hypothesis driven, would be the investigator decides which protein sets to measure, and you avoid this, nuisance marker biasing analysis. And then the data driven approach would rely on each participant as a natural experiment, and you're basically just following that one person, right? So if you're going to do a longitudinal approach in your database, if you have samples from an individual that you're going to follow over time, that's probably going to yield you a lot better than comparing cross-sectional samples any day of the week, especially with inflammatory factors. And if you're going to run a trial where you're starting with a pre and a during and a post, then that's interpretable, because then each person is their own control. And then if you're looking at highly reproducible AD and inflammatory protein families, the idea is to figure out which ones go hand in hand. So in our Parkinson's study, we figured out that there was a really interesting different relationship if you looked at inflammatory factors along with synuclein and along with A-betas, that the relationship between a certain inflammatory cytokine and the level of synuclein and the level of A-betas was different for controls versus case. And so if you look at both the neuropeptides, the neurodegeneration peptides, and the inflammation, you probably get a better measure of differences than if you just look at inflammation in isolation or the neurodegeneration peptides in isolation. And so this is what William feels is a reproducible protein families based on his latest work, and that he presented at the ApoE immunity conference recently that Guojun co-chaired, and he presented a short talk with this. And so basically, by looking at some of these analytes, he was able to sort of stratify or distinguish a little bit some of the different cohorts, and this may be a way to go and get a little bit more granularity potentially. And finally, what he feels is interesting, and I think it's still early days, but he just published this, is this ability to kind of combine, say, one of these markers like soluble TREM2 or soluble TNF receptor with what he calls, high core AD, and combine those two and be able to predict conversion to dementia. So some of those that had like high core AD and low soluble TNF receptor are following this trajectory, versus those that have low core AD and high soluble TNF receptor are following this trajectory. So this is, one cognitive decline from baseline MCI, and this is cognitive decline from baseline AD dementia. And so what's interesting, and I told you the biology of TNF, is that soluble TNF receptor is shed when inflammation is around, right? So presumably its role is, yes, a biomarker, but presumably its role is to try to sop up and neutralize TNF, the soluble TNF that's circulating. So, if you have more soluble TNF around, then you would presumably, that would be an adaptive response to sop up inflammation. And if you have less soluble TNF-R1 around, you would not be as able to sop up that inflammation. So, if that's true, then some other inflammatory analytes should be able to, confirm that, or we should be able to look at some of those, and maybe those will be down too. So the summary then, the take-home message is many inflammatory and non-inflammatory proteins co-vary even when their theoretical functions are thought to be related or independent. Prediction models incorporating multiple such biomarkers risk collinearity. Altered levels of CSF and plasma inflammatory proteins cannot be interpreted at face value according to their in vitro functions. I think Tony has said this before. He hates the word anti-inflammatory and pro-inflammatory. I think it is all context dependent, absolutely. But, you still see these terms being thrown around. Orthogonal protein family measures can serve as biomarkers for AD subtypes, prognosis, surrogates. And then sort of the bigger picture is that, these are definitely not for the faint of heart. You got to think about what biological phenomena are they representing. We know that there are a lot of brain-specific cell types that are being identified by the single-cell RNA-seq that a lot of people are running. We can't just say that, an increase in protein X or analyte X doesn't mean that there's increased glial secretion of it in response to that pathology, because there's a lot of multiple processes going on, and it's difficult to infer exactly what is happening. William feels that these are likely to be identified by novel methods, but not in large consortia. And the other thing that I think is really lacking, and a biomarker core is not likely to make a dent in this, but the NIA probably could, is that we need more knowledge of the peripheral central crosstalk and the two-way traffic between the periphery and the central compartment. And I know they just had an RFA to address that. So, help is on the way. There's significant interest in inflammatory biomarkers, but again, there's limited harmonization. We talked about, some of the issues with assay validation and the different platforms. The problems with, how to control for systemic disorders. Those we know are playing a role, and how do you take those into account? Sort of the big elephant in the room is, the long-term biospecimen stability and the effects of freeze thawing, which, are obvious. And the more aliquots you have, the better, because you definitely don't want to go there, since that will definitely influence. Some analytes go up with freeze thaw and some go down. And then finally, something that's pretty obvious is that it will take a fair amount of personnel time and effort to standardize the fluid collection and assays that will be needed to prioritize all of that. So finally, the goal of distinguishing between AD and controls that we had been doing up until now, I think, has, in my opinion, pretty limited utility. And where we need to focus our effort is in the endophenotyping and trying to figure out, how to stratify and how to be able to use some of these to stratify for trials, so that we don't take all comers. If you take all comers, you end up with a disaster, because even inflammation is not inflammation, right? So you've got to be looking for the right markers and following the right markers to be able to to test the hypothesis. One of the things that is important that William felt is something that needs to be done is that a content expert group should guide the efforts alongside clinicians who may be more prone to want to steer in their own favorite direction based on mass spec, or let's just do postmortem CSF or whatever, in that you tend to go with the expertise that you have in house rather than what may be the best practice across. So I think that's it. I mentioned the lessons from Parkinson's, and I showed you some of the 24-hour sampling. I won't really go into the PBMCs, but just to mention that, when we plate the PBMCs and you can use those to see how inflammatory they are or how phagocytic they are, you get a lot more information. And just imagine that what you're doing is you're leveling the playing field and you're really setting up a controlled experiment, right? So that everything is controlled, and then you hit the go button, and then you can really compare how the delta of activation of each inflammatory trait, not a state that you happen to capture when that patient came into the office. And then finally, something that you don't have in the biorepository, and that's the stool as a peripheral biomarker. So in our interest with microbiome, we started asking the question, even if the patient has dysbiosis, is the patient inflamed, right? Even if they have all these short chain fatty acid producing bacteria that are missing, are they inflamed, or have they compensated for that? So we've been doing inflammatory markers in the stool and looking for zonulin and calprotectin and all these things. And we just published a paper recently in Boost Journal showing that there are some hallmarks of leaky gut in PD patients that may be able to predict some of the trajectories and some of the PD symptoms. And the important take-home message there too is that the plasma inflammatory factors don't correlate with the stool inflammatory factors. So if you want to look for gut inflammation, you've got to look in the gut, right? And so the stool cytokines were very, very telling, and so they're pretty easy to measure as well. And I think I'll stop there.
Thank you, Dr. Tansey. I hope that right.
Thank you for your presentation and your thoughts on inflammation and biomarker outcomes. We have 10 minutes to ask questions, and Bud Kukul had typed in the first question about inflammatory markers and their association with a cognitive syndrome and their association with ATN.
Okay, where's the question again? I'm sorry.
Yeah, it's in the chat. How do inflammatory markers differ in association with cognitive syndrome and their association with ATN?
I mean, it's there's no there are no known and accepted markers of inflammation for cognitive syndrome, or even any that I would call, these are your discriminatory ones for Alzheimer's or ATN or PD. I mean, everything is very variable, and not all patients are inflamed. I think there's some estimates that Michael Hennick and I were talking about the other day. It's like 60 to 70% of patients in any one neurodegenerative disease have underlying inflammation. And in some of them, it may or may not be driving their disease. So there's no, qualified inflammatory biomarkers for any of these diseases. So I couldn't tell you necessarily which ones are definitively ones for cognitive syndrome versus, any one particular one, unfortunately. Did you want me to read the others in there?
Yeah, there are two others from Anne Fagan and Suzanne Kraft.
Can you address the finding of several studies at LA levels and inflammatory markers for gamma attacks and associated with slower progression symptoms? This has been towards respecting a preserved body. Let's make it for resilience and similar was. Yeah. Right. I mean, I think there's a good possibility that if you have more anti-inflammatory or ability to have more reserve, you are able to have slower progression. But I think it's going to be more complicated than that, right? Tony can probably tell you much more about eotaxin than I can, for sure. It's not going to be as simple as anti-inflammatory and pro-inflammatory. If it was, then I think, we could just take a page out of the autoimmune disease and be done with it. One of the biggest problems that we have is that a lot of the drugs that we might be tempted to use will be immunosuppressive. And we don't want to do that to the elderly populations, right? And so that's a problem. I think it's tempting to just go with something, as dirty as, corticosteroids or, even anti-TNF drugs. But I think we have to be really careful and identify the pathways that will affect the arms that are affecting the vulnerable populations and not deliver collateral damage by affecting those that are necessary. So I think it is a plausible hypothesis, absolutely, that something that's more anti-inflammatory, more Th2 type wound healing kinds of pathways are correct and are more protective. Is there another one? Let's see. Thank you. I'd love to hear your thoughts on the impact of chronic lifestyle stressors, how this may impact future studies. Okay. It is clear that stress, chronic psychological stress, and one of my postdocs did a model of chronic psychological stress, CJ Barnum, who is now with Immune Bio, triggers a lot of inflammation in the brain, different in different brain regions. And it's actually worse than the chronic unpredictable stress models that people like to use. And I think that one of the things that we don't know yet is how chronic stress is affecting the gut microbiome and is affecting how we are that central peripheral compartment. And so I would like to think that we need more studies into chronic life stressors, how, being hunkered down at home has affected us, and how lifestyle and exercise are potentially our friends. When people ask me what's the best anti-inflammatory, I always like to say exercise and good diet. And, I think as neuroscientists, we were probably a little cocky in that we didn't realize that what's good for cancer prevention and good cardiovascular health is probably what's going to be good for your brain, which is good diet and exercise and sleep. And so I think, here we are. So I would think that lowering your chronic stress and sleeping and yoga and exercise and the kinds of things that Dr. Yokoyama likes to promote would be a good thing, because I do think they will contribute to chronic inflammation and that sort of chronic innate immune dysfunction that we think is fueling that chronic inflammation.
Thanks. We have the final question from Tony, and he's asking, do you think that gut inflammation triggers systematic inflammation or the other way around?
Systemic inflammation. Yeah. I think it could be both, Tony. And we have some studies with colitis that we just published recently by Madeline Hauser, where we do subthreshold stimuli, and colitis can sensitize to other neurotoxic stimuli, and in a genetically sensitized environment, they can deliver a nigrostriatal hit to in an animal model. So we believe that systemic inflammation could give you leaky gut, and leaky gut could also give you circulating cytokines, through endotoxin. And so it could be, a feed forward cycle. Yeah.
Thank you again for your acceptance of my invitation and coming to give a keynote presentation at this meeting today. I hope that everyone found your presentation very stimulating and helpful. In the next section of the meeting, the topic is vascular biomarkers. We've had some conversations about vascular biomarkers in the past, especially in the clinical course during committee meetings. And so we had two volunteers to come and talk about some of the things that they're doing in vascular biomarkers. And our next presenter is Dr. Mitzi Gonzales from one of our new ADRCs from at South Texas ADRC. And she's going to talk about the overlap of VCID and AD biomarkers.
Sorry, everybody. Can you see my screen? Is that better?
It looks like it's there you go.
There you go.
Okay, perfect. Well, thank you very much. I am very excited to be here today to speak with you on the overlap between VCI and AD biomarkers as a representative from the South Texas ADRC. So we know that vascular cognitive impairment, it refers to the kind of entire spectrum of vascular brain pathologies that contribute to any degree of cognitive impairment. And this can range from subjective cognitive decline all the way to dementia. It is a highly heterogeneous disorder. There's multiple underlying cerebrovascular disease processes that can lead to disparate types of tissue damage and have multiple interactive and downstream effects. In addition, we know that the cognitive, motoric, the behavioral symptoms that are associated with VCI are pretty heterogeneous, and this depends, of course, on the type, the extent, the underlying location of the cerebrovascular pathology. And for this reason, VCI is challenging to reliably diagnose in the clinic and also to systematically study. To make matters more complicated too, we know that cerebrovascular pathology rarely exists in a vacuum. It's been estimated that among adults with dementia, about 70% will have some degree of vascular contributions to their underlying presentation. And the presence of infarcts and white matter lesions, they're not benign. It's been shown that among individuals with Alzheimer's disease who are likely to also have comorbid vascular pathology, that the presence of those infarcts or white matter lesions or other forms of small vessel disease actually contribute to cognitive impairment and the risk of dementia. Therefore, it really highlights the need for us to be able to better understand the epidemiology of vascular cognitive impairment, to be able to reliably diagnose it, both in its pure and etiological forms. This work has been made very challenging by the lack of validated biomarkers. So I'm going to speak today on behalf of several members of our institution that are doing work in this area to promote this line of research. So we are a member of the Mark VCID consortium, which was mentioned earlier, which is comprised of 9 sites and a coordinating center. And its overall goal is to both identify and validate fluid and imaging biomarkers of small vessel disease, with a focus both on the instrumental validation as well as on the clinical validation. One of the markers that's been systematically examined.
I'm sorry, Dr. Gonzales. Can we have you resize your slides because they're not fitting in the frame?
Sure, absolutely. Thank you for letting me know. Let's see. Is that any better? Let me try again. Start from the beginning. Okay, what are you seeing?
Just 1 minute. If you go to the top where it says display settings, there's a down arrow and just toggle that.
Okay.
There we go.
Okay, thank you. Appreciate the technological help. So this is some work. Oh, I guess I skipped a slide. So we've been, one of the biomarkers that's been studied in Mark VCID is peak with skeletonized mean diffusivity, or PSMD. So this is a fully automated imaging measure that's derived from diffusion tensor imaging, and it's believed to capture the diffusion of water, which is indicative of white matter microstructural damage. And it's been shown that in individuals with both genetic forms of small vessel disease like CADASIL, as well as individuals that have a higher burden of white matter hyperintensities, that they have higher values of PSMD, suggesting diminished white matter integrity. And Dr. Claudia Satizabal, who's the leader of our population neuroscience core, has taken the lead on a multi-site instrumental validation study. And across sites, the measure really showed very strong interrelator reliability, as well as test-retest reliability. Another marker that's been explored in Mark VCID is plasma neurofilament light, which of course is a putative marker of neuronal and axonal injury. It's of course considered a biomarker for AD, as well as other forms of neurodegenerative disease, and small vessel disease or vascular cognitive impairment being no exception. So it's been shown that individuals with small vessel disease do have higher levels of serum or plasma NFL. And again, in Mark VCID, a lot of work has been done across sites in order to look at the instrumental validation, which has been shown to be quite strong. In addition, the clinical validation has also been found to be pretty strong, with associations with poor general cognitive function in association with higher plasma NFL. And of course, before we can actually implement and use biomarkers, particularly from blood in the clinic, we really need to be able to understand all of the potential confounding factors. And one factor that's been proposed as something that we should look at is renal functioning, because it's been hypothesized that the kidneys clear NFL from the plasma. And to systematically examine this, Dr. Hugo Aparicio led a study in the Mark VCID Charts Consortium across multiple very large population cohorts, the majority being non-diabetic, and found a very strong association between decreased renal function and lower or higher plasma NFL levels, as you can see in the scatter plot. And this is important because it suggests that there's a risk of possibly overestimating the extent of neuronal axonal damage potentially in individuals with poor renal functioning, and just really points to the need for us to further validate these biomarkers in really large, diverse samples so we can better develop predictive algorithms, considering the complexities that people present with both systemically as well as within the central nervous system. When we talk about generalization too, another point that we really need to consider is our ability to engage diverse populations. We know that the vast majority of work in blood biomarkers has been primarily conducted upon individuals who are non-Hispanic white. So to help fill this gap, we conducted a study within the Texas Alzheimer's Research Care Consortium, and we looked at Mexican American older adults. And at baseline, we assayed several different putative biomarkers for ADRD that are considered markers of axonal or glial injury. And we looked at their ability to predict incident dementia approximately 4 years later. We found that total tau, neurofilament light, and GFAP were all significantly associated with incident dementia later on. However, GFAP, which is a putative marker of astrogliosis and has been associated with both AD and with BCI, was the most robust predictor. And in addition, we found that adding additional biomarkers to the model did not improve the overall predictability of incident dementia. So suggesting that in this cohort that we studied, that GFAP was a pretty robust marker of incident dementia. In addition, other people in our team are doing work to be able to validate markers beyond those that are fluid and neuroimaging. So Dr. Gladys Maestre and others are leading studies to non-invasively assess the retinal microvasculature using optical coherence tomography angiography. It's been shown that the retinal microvasculature has a lot of anatomical and physiological overlap with the cerebrovasculature system, suggesting that this might provide a good non-invasive indicator of the extent of small vessel disease in the brain. In addition, one of the most robust risk factors that's been shown over and over again to be a risk factor for both dementia and stroke is hypertension. But we're all well aware that just a single measure of blood pressure may not be able to adequately capture somebody's typical blood pressure, and more importantly, perhaps that variability in blood pressure. So Dr. Maestre and others are doing studies where they're examining ambulatory blood pressure 24 hours or just at night to be able to see whether this is a stronger predictor of adverse cardiovascular and cerebrovascular events. And then finally, I just wanted to quickly highlight some work too by one of our neuroimaging core leaders, Dr. Mohammed Hobbs. So he has a body of work where he is using artificial intelligence and machine learning techniques to be able to actually develop algorithms that detect specific forms of underlying vascular pathology. So this is really important too to advance therapeutic discovery. We need to be able to reliably detect specific forms of vascular injury. So obviously there's been a lot of work that's been done in this field, but lots more to do. So we need to further our research on instrumental standardization in order to validate biomarkers that are reliable, reproducible, and feasible to implement. We need to further conduct this research in really large, diverse cohorts so that we can develop algorithms that help us better understand the association between these biomarkers and risk for dementia when considering sociodemographic factors, comorbidities, mixed dementia presentations. We need to do more research in order to identify which biomarkers are in fact the most useful ones for predicting cognitive decline, and this might change, of course, across the disease continuum. And then finally, we really need to improve biomarker development that specifically reflects underlying pathologies associated with BCI to be able to advance therapeutic discovery. And as a new ADRC, we're very much looking forward to partnering with you on these efforts. So thank you very much.
Thank you, Dr. Gonzales. We have another 10-minute presentation, and then that will be followed by 10 minutes of Q&A. And so we're going to have Dr. Guojun from Mayo Clinic, ADRC, to present next.
Thank you. Let me try to share my screen.
Success.
Is the screen okay?
Success.
Okay. So thank you for the opportunity. Guojun, Mayo Clinic. I'm actually specific in Jacksonville. So the Mayo ADRC Biomarker Core, we're actually two sides. Mayo Clinic Jacksonville, where I am, and then Michelle Mielke, who's going to give a talk later. She's in Rochester, so we're co-directing our biomarker core. So really the goal is to collect and bank these biofluid samples, generating data set, and then also our center's theme is to address similarity and differences among Alzheimer and ADRD, FTD and DLB and so on. So my own interest has to do with ApoE, which, as you know, is the strongest genetic risk factor for Alzheimer's disease. You know, 2 allele can give you 10 to 15-fold increase of risk. E2 is protective. In terms of biology, ApoE is a lipid carrier, both in the periphery and in the brain, binding lipid in the C-terminal and then delivers through N-terminal binding to ApoE receptors, LRP1, LDL receptor, and HSPG. So ApoE was discovered in 1993 as a strong risk factor. The field had learned a lot how ApoE4 is a strong risk factor. It actually probably the primary pathway is still to influence the amyloid deposition by inhibiting A-beta clearance and increase aggregation. But it has a lot of these other pathways, and many of them are not necessarily related to A-beta. For example, Dr. Zulokbek, who's here, have shown that it increases blood-brain barrier leakage, impair vascular function. Then there are a lot of pathways that ApoE4 impair metabolism. There are also loss of function components, for example, impair the lipid metabolism, and some of that also relate to what Malú had talked about, inflammatory responses. So in terms of cerebral vasculature, this has been sort of talked about in the previous Dr. Gonzales talk. Really, many of these pathological lesions, atherosclerosis, arteriosclerosis, CAA, they can all lead to changes in vascular contraction dilation, and some of them affect the cerebral blood flow. Also in the capillary, I mentioned about blood-brain barrier, but also there could be impairment in vascular genesis, angiogenesis, and really many of these factors impacting Alzheimer's risk or vascular risk really converge in these pathways, aging, ApoE genotype, and sex. So really many of these pathways we think really directly contribute to the development of Alzheimer's disease. So in our lab, we use different model systems to study vasculature. I think I totally agree with what Malú is saying, that the point of doing my biomarker, we should try to understand what's the biology behind or pathobiology behind everything. In this case, these were animal model. We conditionally express ApoE3 or 4 just in the vasculature using the SM22 Curie. So we did the single cell RNA sequencing analysis. You can see we can capture all these clusters that are relevant to the vasculature. And in this simple study, not necessarily simple, but really the outcome, you can see that if you look at the E4 animal, you can see in the vasculature, you have the so-called gliogenesis or gliosis, which is validated in here. Whereas if you express E3 in the vasculature, you enhance the parasite function. In another study, Mayo Clinic, we also have a large brain bank. We look at the correlation of soluble form of different protein that implicate in Alzheimer's disease versus pathology. In this case, we show that the soluble tau or soluble ApoE negatively correlated with vascular marker, which these are tight junction protein, claudin 5, occludin. And then, there are more information in this publication. This is more of a pilot study. This is only N equal 15, 7 male and female, but these are individuals. We have both CSF and plasma. So really the exercise to look at the correlation of some of the proteins we're interested in, including ApoE, and some of the inflammatory or vascular related biomarkers that we use. In this case, we use Luminex multiplex assay. But you can see in these this particular pilot study, some of these inflammation-related biomarker and also the vascular marker really correlated with, highly correlated with ApoE up to myoglobin, Pi1. This happened to be all ligands for LRP1. So CSF, you have very strong correlation, but plasma, not as much. And this just emphasized the point that many of these proteins actually do not cross the 2 domain, so we have to study them separately. And here just some specific correlation, ApoE with ApoM, ApoE with complement factor H, and also soluble VCAM with ApoE. So here's the table summarizing the effect of aging on some of these markers. So you see that many of these proteins go up. The only thing that go down was the plasma complement C3, but everything else is pretty much going up. Well, these are many of these are significant in this study, and this is just a specific correlation you can see as a function of age. Many of these proteins go up, and some of these proteins actually correlate with one another. The other thing I want to mention is that ApoE is also highly expressed in the periphery. E4 not only is a risk factor for Alzheimer's, but also a risk factor for hypercholesterolemia and atherosclerosis. So there's a whole set of biology of ApoE in the periphery. So one of the things we look at, Randy Bateman's lab does this as well, is to use mass spectrometry to quantify ApoE, which is more precise than the typical ELISA. So basically, we both find that the disease status does not change, influence ApoE level in CSF or plasma, but ApoE genotype, E4 actually have lower level in the plasma for ApoE. You don't see that in the CSF. And when Randy Bateman did the stable isotope labeling and turnover study, he found that the E4 actually have a faster turnover in plasma compared to E3 or E2, and this is not seen in CSF as the underlying mechanism. So this is also reflected if you look at the heterozygous individual in the plasma, E3, this is a CSF in 3 or 4, you see E3, E4 is basically 50-50%, which makes sense, right, heterozygous. But in plasma, because of faster turnover of E4, about 70% of the ApoE is E3, only 30% is E4. So this is another study we did just to do a phenotypic correlation. This is a collaboration with Rick Casale at Mayo, Arizona. So basically you can see that the ApoE level in the plasma really contributed by E3, very little by E4, and E3, E4 do not really correlate. But what we found is that the level of E4 to E3 negative correlate with the outcome of glucose metabolism and also the gray matter volume. So the lower level E4 is actually beneficial in this particular study. Finally, just we're doing this lipid analysis, really looking at the lipid level in plasma in relationship of many of these apolipoprotein, not just ApoE, but many of these other proteins as well. This is a busy slide, but simply say that this is a CSF apolipoprotein. You can see that the ApoE, which is this purple line, really overlap with ApoJ, and these two apolipoprotein actually exist in overlapping or same lipoprotein particle. As you know, ApoJ is also known as clustering, which is also risk for Alzheimer's disease. Whereas ApoA1 is mostly on a different particle, also sort of exists with together with ApoA2, one of the less talked about apolipoprotein. And then finally, the plasma ApoE does have an impact on what's going on in the brain. So this is also one of those animal studies where we conditionally express ApoE only in the periphery, bred to the ApoE background, so there's no ApoE in the brain. So if you have ApoE4, you'll see significant leakage of, in this case, the dextrin dye in the brain compared to the glutamate controls. You can also reproduce that by injecting plasma. This is Tony Wyss-Coray, who's also here's really young plasma effect. But here, except we put in the ApoE genotype in here as well. E3 seem to enhance cognition in these older animals, but E4 did not seem to have same effect. And if you look at the blood-brain barrier integrity, E4 plasma actually also cause leakage compared to E3 plasma. And based on these type of animal study, we're actually conducting a clinical trial that really just giving young plasma from E3 individual to individual who are in the early MCI stage of AD, but E4 in terms of genotype. Really, this is a phase 1 study, but we have a lot of biomarker built into that. So lesson learned, vascular and brain ApoE, from model studies, we learned that they do impact vascular integrity and function. Multiple vascular inflammation-related marker in CSF and plasma associated with level ApoE, another LRP1 ligand. Plasma ApoE4 is lower than E3 in the E3 E4 heterozygous carrier. E4 level is associated with hyperperfusion and reduced gray matter volume. So basically also evidence that E4 is somewhat toxic to cerebral vasculature, likely by some type of gain of toxic function, primarily impairing blood-brain barrier. Just want to acknowledge ADRC, Ron is the director. I mentioned about Michelle and I, 2 sites co-directing the biomarker core, but data I showed today, Dr. Connecchio and Dr. Martins contributed. And then much of the work is obviously ADRC support, but we also have some internal funding to enable some of our study. So I think I'll stop there.
Thank you, Dr. Wu, for your presentation, and thank you, Dr. Gonzales, for discussing vascular biomarkers. Excuse my internet problems that I'm having right now, and hopefully there are some questions in the chat, and Grayson will be reading those and presenting them to you. You can find them in the chat as well.
And I think the first question was for Dr. Gonzales. What do you think are the most specific CSF biomarkers for neurovascular disease as opposed to more general glial, amyloid, or neuronal injury pathology?
Yeah, that's a great question and a big challenge for the field because ultimately the downstream effects with the inflammation and the neurodegeneration are really common to both. And so I think that we need further validation to say for sure what's going to be our ideal markers. I know there's some research suggesting that NFL might be more strongly associated with vascular forms of cognitive impairment than AD, but in all likelihood, we're also likely going to need multiple biomarkers because VCI in itself is so heterogeneous and it's different pathologies too. So good question, but one that we definitely need more research to fully address.
Thank you. And I believe this next one is for Dr. Bu. Do you have any thought about the relationship of ApoE4 to AD disease that occurs in different clinical syndromes? For example, amnestic versus aphasia versus physiospatial.
Right, so it's a good question. Actually, an expert on that is my colleague Rick Caselli, who really follow ApoE4 individual longitudinally. And if you discuss with him these different cognitive domain tests that he gives these individuals actually do have different outcome. So I don't have specific answer, but there's recent study actually E4 sometimes can be even beneficial, for example, for some of the specific visual and spatial memory. So I think it's a good question. I think we should all try to follow up and think more about that.
Great, and we have a few more for you. Does the plasma E3-E4 ratio in heterozygotes hold up across racial groups?
Great question. We're looking at that. So obviously we're trying to build a cohort. I think at Mayo ADRC, the Jacksonville site, we do have an African American cohort, and recently we moved to hopefully a Hispanic cohort. So that's exactly the thing we're gonna look at, but I don't have the answer for now.
Okay, and it sounds like we have a question about when you will know the results from the clinical trial treating E3, E4 AD.
So we just enrolled the first patient a couple of weeks ago. I would say probably this is a 12-month trial. So 6 months treatment, either complete plasma exchange or plasma dilution, and another 6 months of observation. So it's a 12 months trial. I'll say probably in the year and a half, we'll have some safety, of course, but also some of the biomarker readout.
Great. And this next one is also for you. Neuroimaging studies have found that young, cognitively healthy ApoE4 carriers have a hyperactivity, hyperconnectivity phenotype. Sorry, I'm scrolling, which some have argued is a compensatory response to maintain cognitive function. From the molecular cellular effects of APO4 that you discussed, can you speculate how ApoE4 genotype may lead to this hyperactivity, hyperconnectivity phenotype?
Right, so another great question. If you actually, I mentioned about Gonzales, if you look at what he had learned, even individual a bit older, let's say 50, 55, and so on, E4 individual actually can have better learning. So if you give this individual every year cognitive test, right, over a 5-year period of time, you see the E4 carrier, their performance actually enhances over the period of time compared to the non-carrier. So I think there are just multiple indications that E4, I don't know if this is compensatory or there are some other underlying mechanism, but there are some, either you can call it advantages in learning or other ways that, E4 is not necessarily always inferior. Really, the risks related to memory and Alzheimer's disease occur after about 55 and 60 years old. And, even in general population, you can see cognition or memory start declining, but E4 carrier obviously decline faster. But at the younger age is a completely different story. I don't think there's enough work been done. I think it'll be interesting to further follow up on a question like this.
Great. And the last question, if you could briefly explain why you did not use ApoE2 mice.
Right. So that's much of that related to funding. You can only do so much. Much of these studies are quite complex breeding. But we do have ApoE2 animal, trail behind. But I just mentioned one or two things if I have a last minute to talk about it. E2 obviously protect against Alzheimer's disease, but it is a risk factor for CAA and CAA-related hemorrhagic stroke. Very evident on the clinical side. So we do have these animals that express E2 in different domains and look into that. But we think the protective effect of E2 in Alzheimer's really related to a better ability to mediate lipid metabolism. E2 lipoprotein typically are larger compared to E3. The E4 is the smallest, coincide with E4 more likely to aggregate. But in terms of in the vasculature, why E2 give you higher risk for CAA, we don't have the answer to that yet. Maybe the mechanism for A-beta aggregation at CAA is somewhat different, but obviously very great. If we learn more about E2, maybe it could be more helpful to think about therapeutic strategy.
Thank you, everyone, for your presentations and for your questions. We split it in a nice 10-minute break right now, so we'll be back presenting at 12:30, so you have 10 minutes, and that's it. Tony, are you online and ready?
Yeah, should I share my screen?
Yeah, you can share your screen. It's 12:30 and we're back in session. And our next 5 presenters have been going to be presented all at once, one right after another. And so if we could have the speakers kind of monitor the chat and answer any questions that are specific to you, that would be great. But we still will have a 10-minute Q&A at the end of the 5 speakers. Our 5 speakers this afternoon are Dr. Tony Wyss-Coray from Stanford University ADRC, Michelle Mielke from Mayo Clinic ADRC, Wendy Q from Boston University ADRC, Thomas Wisniewski from NYU ADRC, and Julio Rojas from the University of California, San Francisco ADRC. So thank you all for volunteering to present, and please start, Tony.
Thank you, Cerise. Thank you so much for giving me the opportunity to talk about our work. And this is a sort of a work in progress that I'm going to present to hopefully stimulate some ideas and discussions. I'm representing the Stanford Alzheimer's Disease Research Center, which is directed by Victor Henderson and co-directed by Kati Andreasen. And in the biomarker core, we're directing this together with Kati Andreasen. Nima Agepur is a computer scientist. Divya, Patricia, and Malia are helping us direct this core and run it. And it's really thanks to all of them that we have been able to collect a lot of samples and do these biomarker studies. So our principal question, and Malú has discussed this already, is to really find biomarkers of dementia and of brain aging. And the question is, can we do this without accessing the brain? Because of course we can't get biopsies from the brain. And the idea is that the cells that reside in the brain communicate with the vasculature and release proteins, factors, other molecules into the blood that will allow us to get a snapshot of what happens in the brain. And so what's in a drop of blood? We, of course, all collect now plasma. Some people still collect serum, but plasma is without coagulation. You collect the liquid fraction. And in this liquid fraction, you really find a plethora of different molecules, tens of thousands, that are not all soluble. Many are actually associated, as Guojun just described to us, in lipoprotein particles. Other are in different types of vesicles, exosomes. And then you have soluble proteins. Some are in free forms, are actually in aggregates, and lots of other proteins and molecules. So the question is, can we gain information about not just the whole body, but of specific tissues such as the brain? And because age is the key risk factor for cognitive dysfunction, neurodegeneration, Alzheimer's disease, we reasoned that if we can understand the changes that occur in the blood with aging, it may teach us something about aging of different tissues, including the brain. And so we set out a number of years ago, and this is published since, in trying to get ever increasing numbers of proteins measured. And this is a culmination of a long study with Nir Barcela, Sophia Millman at Einstein College, Adam Butterworth of the Interval study, and particularly with Somologic, who have developed this platform that now measures over 7,000 proteins with these so-called aptamers. These are oligonucleotides, modified oligonucleotides that specifically bind a protein. They can be synthesized, so they're very highly reproducible measurements. And again, we can now measure with this platform almost 7,000 proteins. Here we had samples from 4,000 people, healthy people age 18 to 95. We measured roughly 3,000 proteins. And we can show these dramatic changes that occur with age. So here you have age, you have the 3,000 proteins for each person, and the 4,000 people here in the x-axis. And you can see that proteins are sometimes low in blue when you're young, and they increase with age, and vice versa. Lots of changes happening with age. Because of that, you can actually model the content of the changes with age, and you can build these so-called clocks. So here we can calculate, based on the concentration of these proteins, we can basically calculate the relative age of a person. This is often referred to as biological age. And it's, of course, particularly interesting how people deviate from this perfect prediction of the actual chronological age in time. So we call this delta age. So if a person is predicted to be younger, are they biologically younger? Are they functioning better? Is their brain functioning better, and vice versa? And this seems indeed to be the case, and many other groups have tried to develop such clocks. But the challenge with them is that proteins are derived from any tissue or cell in the body. So we don't really know what exactly you're measuring. And these are, I would say, organismal clocks. So they give you a relative understanding of the overall age of an organism. But we know now that tissues age at different rates, cells age at different rates. And you really not want to know how old is the liver, how old is the brain, how old is the heart in a given person. And so to do this, what Jared Rutledge and Hamilton O, two grad students in the lab, decided to do is to figure out which proteins of these 7,000 that we can measure now are from a specific tissue. So many proteins are derived from many different cell types, or even from every cell type that you find in plasma, but some are unique. And we derive this uniqueness or specificity from gene expression data sets. So if we see that a gene is only expressed, let's say, in a neuron, we consider the protein derived from this gene to be brain specific and so forth. And what's really encouraging is that if you look at these 7,000 proteins, a few hundred are actually derived from the brain, some from the liver, blood, and so forth. So can we then build tissue specific clocks that tell us, for example, how old the brain is? And indeed, when we look now again at this prediction of the overall organismal clock I showed you before, we get an R value, so a correlation prediction value of about 0.9. But even with the brain, we get still a point above 0.8 correlation values, and you can see here for these different organs. So there's a lot of information in these plasma proteins that allow us to predict the relative age of a tissue in a living person. And the question is, of course, are these related to function, and can we potentially use that to monitor brain function or development of cognitive impairment? And so here I just show you a couple of vignettes. This is a heart clock. It uses only 1, 2, 3, 4, 5 proteins actually. And in an unbiased data set, this clock can tell us how old, whether a person has had a heart attack with a very high confidence. Also in a second cohort here, whether they had a heart attack or any other heart dysfunction, actually we can predict with this clock. We can also, apologize, my computer is jumping around here. The organismal clock is not as good as this one, but what is more exciting even is that we can do similar types of predictions for the brain. So the brain clock has a highly significant prediction of whether a person has Alzheimer's disease, but more importantly, if we predict, for example, changes in CDR sum of boxes, we get a very promising hazard ratio or prediction of whether a person is going to have a change in cognitive function. This is just one example. We have multiple other ones. Interestingly, the heart clock also has a pretty good prediction of cognitive dysfunction, but then some other proteins are clearly not able to do this. And also the organismal clock is not significantly predicting whether you have cognitive decline. And of course, we can then ask, what are the proteins that make up this brain clock? And many of them are synaptic proteins. Others are involved in neuronal projections and so forth. So again, this is still work in progress, but it suggests that we could select for specific proteins that are synaptic or neuronal derived, and in this way make biomarker models that allow us to predict cognitive decline and dementia. Thank you.
you want me to go ahead?
Yes, that would be great. Thank you.
Great. Let's see here. Hopefully you can see this. So it's a great talk, and I'm gonna switch topics now to start to talk about plasma p-tau, and really to consider previous discussions talking about the utility of plasma p-tau and how we can bring it into the clinic and some factors that we need to think about. And I think even from the ADRC perspective, I'm not talking about primary care here, but if we do talk about return on results down the road and p-tau becomes a clinical test, some factors we may need to consider in terms of the interpretation of the data. And my disclosures. So multiple studies have shown the potential utility of plasma measures, both of phosphorylated tau species, as well as amyloid beta, NFL, and now also GFAP. However, few studies have examined the utility and accuracy of these plasma markers in a population-based study. Most studies to date have been focused on specialty clinics, typically of younger populations. And so we don't really fully understand how these these markers work in community-based studies, particularly in older individuals, say 80 and older with many comorbidities. So with a move towards these blood-based markers as screening and diagnostic tests, there's really a need to understand how they perform in the general population. And of course, this will be really important for establishing reference ranges and thinking about cut points that can be used in the clinic. So our aim was to identify factors that could affect the levels of plasma p-tau in the general population. So here I'm going to focus on p-tau 181 and p-tau 217. We do have a paper that was recently published in Alzheimer's Dementia a month or so ago, led by Jeremy Sirjanen from our group, who focused on amyloid beta 40, 42, NFL, and total tau as well. In this particular study, we had over 1,300 Mayo Clinic study of aging participants, the majority of which were cognitively unimpaired, median age of 67, but an age range of 30 to 98. All participants had fasting blood draw in the morning. The blood was centrifuged and immediately aliquoted and stored at -80. These samples had not gone through a freeze-thaw cycle. And again, as I mentioned, we examined here, I'll show the data for plasma p-tau 181 and 217, and these were measured on the MSD platform in collaboration with Jeff Dage when he was at Lilly. So one of the first things that we wanted to take a look at, simple to start out with, but how do the plasma p-tau measures vary in relation to age? And so 181 is on the top and 217 is on the bottom, and they're essentially the same. But we can see that starting around the age of 65 or so, there is a sharp increase in plasma p-tau levels. However, when we stratified by amyloid PET status, and the vast, I think about 90% of this cohort had concurrent amyloid PET status, we find that this increase is really only among those that were amyloid PET positive. There is somewhat of an increase among the amyloid negative, but these results really, again, firmly suggest that this is really an Alzheimer's disease related marker. Next, we abstracted many medical conditions from the medical records and also looked at other behavioral factors to try and determine, again, what factors might be associated with the levels of p-tau in the blood. Again, on the left is 181 and on the right is 217. They're generally the same, so I'm not going to differentiate the two. In black is univariate analyses, and in red, we adjust for age and sex. And just to kind of get us started in terms of a quantification, first I want to highlight the difference between those individuals that are amyloid positive and amyloid negative. So I can't fully see this on my screen, but I believe for 217, the difference between amyloid positive and amyloid negative is about 0.7 here. And so when we start thinking about this difference in terms of the context of comorbidities, we find that particularly individuals that have chronic kidney disease, stage 3 or greater, have much higher levels of plasma p-tau compared to those that don't. And the difference between those with versus without a CKD is almost the same as those with versus without elevated brain amyloid. In further work, both stroke and MI were associated, or myocardial infarction were associated with higher levels. And we did see a trend as well for a higher BMI being associated with lower plasma p-tau levels. And this is consistent with the literature looking at blood NFL, in which they also find that higher BMI is associated with lower NFL levels in the blood, presumably because of a higher blood volume for those that have a higher BMI. In some additional work now, we've gone on to start looking with CKD, focusing more on creatinine levels. And it is interesting when we start looking at neural path, we have identified some individuals that have very high p-tau levels, but have low to intermediate Alzheimer's pathology. And when we pulled out their creatinine levels, those that had the really high p-tau levels had creatinine levels of, I think one was 2.4 and another one was 3.8. And so clearly that was driving the elevated p-tau levels as compared to Alzheimer's pathology. Next step was to think about how this might impact cut points. And so we developed cut points that were determined by defining an abnormal range as 1.696 standard deviations above the mean in participants who are cognitively unimpaired and also amyloid negative. So first we developed the cut points based on including or excluding those with stroke, myocardial infarction, or chronic kidney disease. And then we also included those individuals. And you can see that there is a difference, particularly for p-tau 181. So excluding these individuals, cut point was 1.57. When we included them, it was much higher, 1.75. However, there was little difference in terms of p-tau 217. We do find that by using the cut points after we exclude those with these comorbidities, the AROCs were higher. And we also had better negative and positive predictive value. So, I mean, this is just the beginning in terms of showing some important factors that affect plasma p-tau levels. And many of these also affect, for example, plasma amyloid beta levels. It's important to go forward in terms of thinking about these factors in terms of physiological versus risk factor, because if it's a risk factor, that could be a reason why you have the elevated p-tau levels. Something but like BMI in terms of relationship to blood volume or kidney disease in terms of protein clearance are going to be more important, I think, to think about in terms of developing the reference ranges and cut points. And we do have ongoing work. I didn't have time to present today, but where we are trying to understand too, for somebody that newly develops or incident chronic kidney disease, how do the levels change in relation to somebody that doesn't, or is there an abrupt upswing? Does it take a while for those p-tau levels to increase? We certainly need to examine these markers in the community and among more diverse samples. And again, I think it's really important to think about interpretation of those results, especially when we're thinking about race ethnicity. So we know African Americans have a higher prevalence of chronic kidney disease and of stroke and some other factors as well. So as we're thinking about some of these comparisons in terms of ethnic racial differences, we need to make sure that we take into account the differences in the prevalence of the comorbidities and not just focus on the racial ethnic differences without that explanation. You know, going forward, this is just for 2 different p-tau assays and isoforms. I think there's certainly going to be a need as we examine others in terms of 231 or other assays. Presumably the physiological risk factors or physiological factors that affect the levels won't change across assays, but still important to consider nonetheless. And we'll also have to think about, you know, doing this in terms of thinking about for both screening as well as for diagnosis, and then whether it's used in primary care or clinical care. So, you know, I think overall, the p-tau and some of the other blood markers are extremely promising and you know, will be going into clinic, I think, sooner rather than later, particularly, and some have already been in clinic. But there are a lot of things that we do need to consider, particularly when we're trying to consider these markers in primary care. So before we go forward, in terms of thinking about educating the primary care physicians, as well as educating other physicians as well in terms of what to look for and how to best interpret these markers. So I really want to thank my co-authors and also Jeff Dage for running these samples for us, and I'm happy to take any questions. Thank you.
Hi, Wendy. If you started, you might be on mute.
Wendy, you need to unmute yourself.
Okay, how about now? Can you hear?
Excellent.
Okay. I'm going to talk about relationship between peripheral chronic low-grade inflammation and AD risk in ApoE genotype-dependent manner. This slide shows BU ADRC structure, the biomarker core through collecting the fluid biomarkers and brain imaging biomarkers. We have been focusing on 3 aspects: peripheral immunology, vasculature, and the brain pathology for AD risk. We're collaborating with NAC, with NICRAD. Locally, we're collaborating with Framingham Heart Study. This slide shows we used the Framingham Heart Study cohort, and we found elevated blood C-reactive protein CRP increased AD risk only in ApoE4 carriers, but not in ApoE2 or ApoE3 carriers. The right panel shows among ApoE4 carriers, those who did not have repeating elevated CRP, the blue line, had similar AD instance as in ApoE, as the one in ApoE3 carriers. This slide shows we used ADNI study cohort, and we divided the participants into 3 groups based on ApoE4 allele, 0, 1, and 2. And we found that higher serum CRP, lower mini-mental scores, only in ApoE4 carriers, at baseline 12 months and 24 months. But this relationship we didn't find in ApoE4 0 and 1 groups. Similar relationship also found between serum CRP and CDR scores. Next slide shows that we found higher serum CRP is associated with increased CSF total tau, only in ApoE4 carriers at the baseline 1 year and 2 years. However, in the absence of ApoE4 carrier, you can see the relationship between serum CRP and the total tau is negatively associated. And in the relationship between serum CRP and p-tau in CSF is negatively associated only in ApoE4 negative group. This slide shows after adjusting for age and sex and education and other confounders, these relationships remain. Okay, this slide shows ApoE2 carriers had the highest concentration of serum CRP, followed by ApoE3 carriers, and that ApoE4 carriers had the lowest concentration of serum CRP. Similar trend was observed in ApoE4, not human ApoE4 knock-in mice. I want to emphasize here, as I just showed you, that elevated serum CRP increased Alzheimer's disease risk only in ApoE4 carriers. However, here I'm showing you the data that ApoE4 carriers had the lowest concentration of blood CRP. So at that time, we had hypothesized that it's different from blood CRP. ApoE4 brains had more CRP stuck in the brain. To approach this hypothesis, first we used purified CRP. It's a pentamer protein and then dissociated into monomer during chronic stage of inflammation. And then we injected them by IP injection into different ApoE knock-in mice. And indeed, we found ApoE4 mice had most CRP, especially monomer species, deposit in the brain. Because most blood proteins exposed to endothelial cells of blood vessels, we further hypothesized that some endothelial surface protein bind to CRP to mediate the pathological activities in the brain. This slide shows after IP injection of MCRP, we found that MCRP co-localized with 3 biomarkers of endothelial, CD31, CD144, and lectin, but not co-localized with markers of neurons, astrocytes, and microglia. We further isolated microvessels from these brains, and we found ApoE4 capillaries had higher level of MCRP and more CD31 proteins are phosphorylated than ApoE2 capillaries. We also used proximity ligation assay and found that after MCRP increased injection, that only ApoE4 brains had binding of MCRP and CD31. This slide shows after IP injection, MCRP reduced capillary length most significantly in ApoE4 mice, followed by ApoE knockout mice, and then ApoE3 mice. However, in ApoE2 mice, MCRP didn't affect capillary length. We also found MCRP increased the level of VWF in ApoE brains, only in ApoE4 mice, and the level of VWF is negatively correlated with cerebral vascular length in the brain. This slide shows we found peripheral MCRP increased the T lymphocyte migration from blood to the brain only in ApoE4 mice. And we found also MCRP induced neuroinflammation marked by increased expression of CD68 and IBA1 only in ApoE4 mice. This slide shows peripheral MCRP increased tau phosphorylation only in ApoE4 cortex and hippocampus. This slide shows we added MCRP into primary brain endothelial cells and found that MCRP and CD31 are co-localized after we knock down CD31 by siRNA. The binding of MCRP to endothelial cells is dramatically reduced. We conducted RNA sequencing and found that CD31 and ApoE are involved in vascular development pathways, leukocyte migration, and RAS protein signal transduction. These pathways under the attack of peripheral MCRP increased in ApoE2 mice, but decreased in ApoE3 mice, more decreased in ApoE4 endotheliums. Here, this slide shows, we go back to human brain tissues. Here we found that AD brains had higher level of vascular MCRP than control brains, and also higher MCRP CD31 co-localization than control brains. And we also found AD brains had higher PCD31 levels than control brains. Next slide shows, in contrast, we found control brains had higher level of vascular ApoE levels and the co-localization of the binding of CD31 and ApoE than AD brains. We injected MCRP and found under this MCRP injection, ApoE2 mice had higher levels of ApoE CD31 binding, but not in ApoE3 and ApoE4 mice or wild type mice. And we found ApoE CD31 binding is negatively correlated with MCRP CD31 binding. So in summary, I think our study showed a possible AD pathogenesis in ApoE4 carriers because during peripheral chronic low-grade inflammation, MCRP bind to CD31 causes cerebral vascular damage and induce T lymphocyte migration and leading to neuroinflammation and neurodegeneration. However, in ApoE2 carriers, because MCRPs hardly bind to CD31 and the brains are protected. And finally, I want to thank my colleagues and the team members at Boston University, especially Dr. Qiushan Tao and Dr. Linda Zhang. And these are also funding resources for our study. Thank you.
Tom, you're muted.
Can you hear me now?
Yes.
Okay. So thanks for the opportunity to speak today. I'll be discussing some of our data on plasma biomarkers of neurodegeneration and neuroinflammation in COVID patients. So these are some of the stark numbers of the pandemic. We're reaching almost 750 deaths in the United States, and we've just surpassed—
Tom, we don't see your slides. Sorry.
Do you see them now?
Yes, thank you.
Okay. These are the numbers. In order for SARS-CoV-2 to infect a cell, there's a requirement of expression of a number of cell membrane proteins. And these proteins are indeed expressed in cells of the central nervous system. The virus can enter the CNS and brain through a number of non-mutually exclusive pathways, which are illustrated here. In the some 200 neuropathological published examinations of COVID-19 patients, certainly PCR has detected the virus in the CNS in some of these cases, but typically it's been at low levels. Much more commonly, one sees extensive microgliosis and astrocytosis, evidence of intravascular thrombosis, and endothelial and blood-brain barrier damage. Sometimes there is a perivascular cytotoxic T cell infiltration. Many studies have clearly shown evidence of microvascular injury, such as this New England Journal paper by Lee et al. Showing breakdown of the blood-brain barrier with extravasation of fibrinogen into the brain parenchyma. A much more recent study has shown that one of the proteases of the virus cleaves NEMO, an essential modulator of NF-kappa B, and ablation of NEMO leads to the death of brain endothelial cells with the appearance of empty basement membrane tubes that one can see in autopsy brains of COVID-19 patients. There is a higher number of caspase-3 positive vessels, and one can recapitulate these findings in animal models. We have conducted a prospective study of neurological symptoms in hospitalized COVID patients in the NYU hospital network. And these patients were from the first wave of COVID from March to May. These are the inclusion and exclusion criteria listed here. Over this period, some 35% of the total admitted patients, some 4,500 patients were COVID positive. 14% of these hospitalized COVID patients had onset of new neurological events. And these folks were quite well characterized. Each had evaluations by neurology attendings. By far and away, the most common new neurological symptom was toxic metabolic encephalopathy, followed by a number of other neurological new events. The toxic metabolic encephalopathy was most commonly due to septic encephalopathy, also frequently hypoxic ischemic encephalopathy, and many patients had a combination of more than one cause. TME was much more common among older patients, and pre-existing cognitive symptoms greatly increased the risk of developing a toxic metabolic encephalopathy. Of the peripheral biomarkers, TME was associated with elevations of IL-6, CRP, D-dimer, and ferritin. We have 6-month outcome measures of these hospitalized COVID patients. And in fact, both groups, both with and without neurological symptoms, fared poorly. Some 50% had impaired cognition and limited activities of daily living. However, those with neurological complications fared even worse, with worse modified Rankin scores and a much lower probability of returning to work. So among these individuals, we looked at various plasma biomarkers using Simoa assays, and these various biomarkers we were able to measure. We obtained serum samples from geriatric COVID-19 patients all over the age of 60 and divided them roughly equally to those with and without neurological symptoms. And they were age and sex matched, with the average age being around 70. This shows the breakdown of subjects. This particular study is distinct from other studies looking at some of these biomarkers in that we looked at a far greater number of subjects. This was limited to geriatric individuals, and we very carefully excluded subjects who had any sort of past history of cognitive impairment or dementia. And then we looked at a more comprehensive set of biomarkers. The patient characteristics in terms of COVID severity symptoms and the type of neurological symptoms is listed here. By far and away, the most common neurological symptom among the folks who had new neurological events was toxic metabolic encephalopathy, present in some 68% of those with a new neuro event. The levels of the individuals with toxic metabolic encephalopathy and new neurological symptoms were very significantly elevated, in particular total tau, p-tau 181, GFAP, and neurofilament light. These levels were almost double or exceeded being double. These correlated with an increased probability of death and a far lower possibility of being discharged home. Amyloid beta 40 and 42 elevations did not meet significance. However, the p-tau 181 A beta 42 level was significantly elevated in this cohort. Importantly, we compared these elevations of neuroinflammation biomarkers to our NYU ADRC cohort of individuals who were normal MCI or AD who did not have COVID-19. And there were very striking elevations in the COVID population. Neurofilament light levels were almost triple in the COVID population, and for GFAP, almost double. This slide shows a heat map of the Spearman correlation coefficients, with greater correlation being shown in green. So these elevated biomarkers correlated with increasing age. They correlated with the severity of the COVID-19 infection. And in terms of the inflammatory systemic biomarkers, there was correlation with elevation of D-dimer levels, perhaps correlating with the prothrombotic state and endothelial and blood-brain barrier cell damage. Hence, with COVID-19, there is very commonly neurological manifestations which occur with a greater frequency in individuals with baseline dementia. In cognitively normal individuals, at baseline, there's significant elevations of neurofilament light, total tau, GFAP, and the AD more specific biomarker of p-tau 181. These elevations correlate with more severe disease and worse outcome. Hence, our findings suggest that individuals with COVID-19 infection with hospitalization may have an acceleration of AD and ADRD symptoms. However, certainly longitudinal follow-up of these individuals is needed. Many individuals were involved in this study, including in the NYU ADRC team, as well as in the NYU Neurology and Radiology Department. The initial funding came from an administrative supplement, but we put in for longitudinal studies in a new grant. So we're hoping to get UDS data and long-term imaging studies on this cohort to work out what the long-term consequences are. This shows a picture of some of our NYU ADRC members from happier pre-COVID times that perhaps we can return to in the next year or so. Thank you.
Julio, are you able to share your slides?
Not yet. I think the other slides need to be taken down first. I'm still not able to share. Okay. Thank you. I will address some of the progress in blood biomarkers in the field of frontotemporal lobar degeneration research. The first idea is that the progress we're seeing, as in any of the other fields that we've been discussing today, is that access to this ultrasensitive technology is leading the way to have a better understanding of the fluid biology in FTD. We went basically in the span of 40, 50 years from ability to measure immunoglobulins and albumin to measure molecules that are present in blood in very small concentrations, but that have a very important role in frontotemporal lobar degeneration pathophysiology. Currently, we're using fourth generation technology based on immunoassay principles, and this has been revolutionary for the field. We have other methodologies that use automated panel approaches. So this will be also part of a revolution. We can also do multi-analyte profiling. We have new technologies that allow screening at high number of analytes in plasma and CSF, and then immunoprecipitation mass spectrometry has also played a role, in particular in the use of the measurement of amyloid in plasma. So blood neurofilament has really been a marker that has changed the perception of how we can understand FTLD using biomarkers. Basically, in the span of the last 30 years, from the first time when neurofilament was detected as relevant in the pathophysiology of FTD in animal models, we are now able to detect neurofilament in plasma. And the most important developments have been in recent years, in the last 5 years, when elevations of neurofilament have been detected in pre-symptomatic carriers of FTLD causing mutations. This has been done through the work of the GENFI, the European Consortium for the Study of FTLD, and then later on with all FTD. More recently, we were able to combine data from all FTD in North America and GENFI to study the trends of neurofilament in asymptomatic carriers. FTLD being an ideal population to study neurodegeneration because mutations have an important component, or they play an important part in the prevalence of the disease. So having an asymptomatic mutation, population is important to understand. So we know, based on the studies from GENFI and all FTD, that neurofilament levels, a marker of degeneration, which is non-specific, by the way, it increase significantly when the full phenotype is present, with less elevations in prodromal stages of the disease. And basically, in normal asymptomatic carriers, the average is that there are no difference compared to non-carriers or normal non-carriers from the same families. However, if you notice in this graph, in normal non-carriers, in carriers in MCI or the prodromal stage, there are some individuals who show elevations, yet they don't change their phenotype. So those, we understand that those individuals may be at risk of progressing in the short term. Those patients or those individuals may be ideal candidates for intervention studies, for trials. So sure enough, people who convert in the short term have higher concentrations compared to non-converters in the asymptomatic population, but also this is true in the MCI or mild behavioral impairment who later progress to dementia. And then GENFI also did a very interesting longitudinal analysis of plasma, and it's those converters, within 5 years, those people who convert, who have the sharpest rise in neurofilament blood levels. showing, and this is across all the main mutations, C9, granulin, or MAPT, showing that this stage of conversion, that neurofilament is really reflecting the disease activity. Also, you see in the symptomatic stage, there's also a rise in levels with time, but it's not as significant as with converters. So this is very valuable information to potentially use neurofilament as a way to identify individuals at risk and to track disease response, the effects of drugs in clinical trials. We have many different variables that correlate with the baseline neurofilament levels. This is a marker of disease severity in FTLD, CDR, NAG, FTLD, sum of boxes. And you see that individuals that have high levels in plasma of neurofilament at baseline in this asymptomatic stage show decline. And it's very easy to separate or to predict who will have that decline based on the concentrations. We were able to replicate this finding in both all FTD cohorts and in GEMFI as well. The third idea, very important, since we've been talking some of the talks about AD biomarkers and the role of AD biomarkers in different conditions. In FTLD, they may play a role as well. We recently published this work where we also collaborating with Eli Lilly, we measure concentrations of p-tau217 in a large cohort of patients with AD and FTLD. And the basic concept is in AD, you expect to see increased levels of p-tau217 with established full phenotypes, and that correlate really well with positive amyloid PET. And there are no significant elevations in the FTLD in total or in looking at the individual phenotypes. However, in those individual phenotypes, there are some individuals who show increased concentrations of the marker, and it's regardless of the phenotype. So the idea here is that this marker could be used to either identify co-pathology, which we know is a white elephant. We know exists, is very prevalent, but it's hard to measure. So these markers in blood may have a role in identifying co-pathology in FTLD, or selecting those cases that when you are presumed to have an FTLD substrate, pathological substrate, you might reveal those cases that are actually AD, and there are mimics of the pathology. So another interesting work by the group in St. Paul Hospital in Barcelona, they have used plasma biomarkers to classify patients in the ATN system. For example, in the FTLD cohort, they use, this is a clinically determined cohort, they can identify based on either a composite of plasma biomarkers, plasma p-tau181 or neurofilament. They can discern who is either A positive or A negative, T positive or T negative, or N positive or negative, based on CSF biomarkers or mostly CSF biomarkers in the study. But you see that within the FTLD group, you can discern who is positive or negative. And here, the attention I want to show you the positive amyloid positive, tau positive are in triangles, and then the diamonds are amyloid positive, tau negative, and you see that those individuals have higher levels in all the different markers. So this may be a new way to characterize our patients as well. For IDEA, we have new biomarkers. The discovery efforts are showing us new candidates, and this is a compilation that was put together by the George Orr group that shows in different biological pathways markers that have been either elevated or decreased in FTD. And there's more research that needs to be done. Most of these markers have been studied in CSF with maybe a couple of exceptions, GFAP, neuropentraxin, but it's an exciting time. Discovery platforms are leading the way into helping us understand and hopefully find, the holy grail in FTD, being able to differentiate tau versus TDP, being able to differentiate FTD from AD with high accuracy. My last idea is that we have a responsibility to care about inclusion, diversity, and equity in biomarker research. We are in a strategic position to facilitate this, to generate research that is generalizable and that looks into the problem of differences in race, even though race is a social construct, but some genetic variables co-segregate with race. And this is not only a matter of race, but also we have responsibility to decrease the gender or the sex data gap, not by correcting for sex, but generating data for sex, for men and women. Thank you.
Thank you. Thank you for all 5 of our speakers on various different topics to present your topics. We have about 5 minutes for questions, and Dr. Rojas, there's a question in the box for you, and it says, is there any blood-based biomarker for sporadic FTLD? Is also neurofilament light generally up in these patients? So if you could answer that. You might be muted if you're talking. Dr. Rojas? Okay. Oh, you muted yourself again. Did you get the question? Okay. I'm gonna move on. There is a question. Yeah, go ahead.
Yeah, I'm sorry, I missed the question because I got disconnected.
Yeah, the question was, is there any blood-based biomarker for sporadic FTLD and also neurofilament light generally up in these patients?
Right, sorry, I was addressing that in the chat. There is no specific test for sporadic FTLD. However, the elevations that I show in genetic FTLD are similar in sporadic FTLD. We also published a recent path-confirmed study in sporadic FTLD, and there is non-specific increase of NFL in FTLD. When you combine with some other biomarkers, for example, phospho-tau and a ratio, you might be able to have certain level of the discrimination between tau and TDP, for example. And you can get to some level of discrimination also using combining with biomarkers with FTD, discriminating FTD and AD. But yeah, none of this is very specific.
Thank you. And then I scrolled higher. John Chow had a question for Michelle, I believe, where he was comparing the earlier presentation of before you, where it showed a strong correlation between plasma, neurolite filament, and GFR, suggesting a renal clearance effect on both neurofilament light and p-tau. And has anyone looked at urine p-tau or neurofilament light? So I think I guess that's a question for all 5.
I'm not aware of anybody that has looked at urine measures of these, and I think it's beyond NFL and GFR. We also see elevations in plasma amyloid beta as well. But again, I'm not aware of anybody measuring it in urine.
And Mary Sano wanted you to clarify whether it appeared that the separation of p-tau occurs around 70 and older.
Sorry, just bringing up the slide. So it starts to increase when we look at everybody without stratifying by amyloid PET status. It kind of starts to increase 65. It goes up certainly the 70 to 74. So I mean, I don't have a specific age that you see the cut point, but it really starts to increase around those ages.
Okay, thank you. And I don't see any additional questions in the chat, but of course that could just be my internet. So we're going to close the Q&A for this section. And again, I thank the 5 speakers for sharing their research. The next section of this meeting is going to talk about logistical challenges and a few more updates that everyone should know about. This will include Dr. Schindler from Washington University and Dr. Farood and Dr. Dage from the Indiana University School of Medicine. So Suzanne, if you're ready to share and unmute yourself, you could do that and we could get started.
All right, can you hear me?
You have to go into display settings and switch your screen.
Okay.
It's in the upper left.
Their screen here.
I think we see your second screen now.
Okay.
And as opposed to the slide deck. And now we see the slide deck and it says next steps. Yep.
Okay. Do you see the top slide now?
Yes. Thank you.
Okay, great. So I'm going to talk briefly about the value of combining data sets across sites and also some of the challenges of achieving this. So as a field, we have now developed multiple biomarkers that reflect Alzheimer's disease brain pathology really quite well. But one concern is that in our interpretation of these biomarkers, we have typically been using a single cutoff to dichotomize individuals as either biomarker positive or biomarker negative. And often we define these cutoffs in cohorts of healthy, well-educated, non-Hispanic, white research participants. And as Dr. Mielke and others have so nicely described today, we have really learned a lot about the fact that biomarker levels are that are associated with brain amyloidosis and with cognitive symptoms vary by lots of different factors, including age, sex, education, race, ApoE genotype, metabolic factors, and comorbidities. So there's a lot of different things that affect these levels. So as we're getting more sophisticated with all of our biomarker analyses, I think that the next step is really to move beyond a single biomarker cutoff that's applied to all individuals and to more precisely determine the biomarker levels associated with amyloidosis and or cognitive symptoms, really for each person based on their individual characteristics, basically to find a cutoff that's relevant for each person. Now, we've essentially already been doing this for a long time by controlling for covariates, but that's kind of been under the hood. And now groups are getting more interested in creating models that are explicitly looking for these effects. And one issue is that these models require large data sets and ideally a diverse population. So if the fuel for creating these models and more sophisticated biomarker interpretations is large data sets, how do we facilitate creation of these data sets? Now, as a community, we're really fortunate to have a lot of data already from many individuals, but unfortunately, we can't just lump all the data together. There are currently a number of studies underway that are attempting to combine data across sites, and as some of you may know firsthand, this is not easy. I'm just going to focus on fluid biomarkers the rest of my presentation, but I think that people trying to harmonize data for imaging and other measures are dealing with some of the same sorts of issues. So with respect to fluid biomarker data, there's quite significant differences in sample collection and processing. Interestingly, some of these differences are a lot more relevant to some biomarkers than others. So for example, the A-beta 42 to 40 ratio has been shown to be less susceptible to differences in pre-analytical factors as compared to A-beta 42. There's also major differences in biomarker assays. Assays for the same analytes may detect somewhat different species. So they're essentially assays for different molecules. They're highly correlated, but they're different molecules. And there's also issues related to lot effects, where the value we get for a sample today is different from the value that we get a year or two later. Now, some of the new assay platforms that are available, like the Roche Alexis assays and the Fuji Rubio Lumipulse assays for CSF biomarkers, have shown in a number of studies that they have good precision and stability. And a number of our groups are using these assays, but some of them are using the Alexis assays and some of them are using the Lumipulse assays. So one thing that we've been interested in is, can we combine data across two high performance platforms? So here is a head-to-head comparison of Alexis and Lumipulse values for 842 CSF samples from the NIGHT ADRC cohort. And for comparison, I would just note that when we ran 29 samples representing a variety of biomarker levels on Lumipulse assays a year apart, we got really high correlations, so Pearson correlations of 0.98 or even better. So with the same assay platform, you measure the same sample twice, you get very highly correlated values. However, if you look at the correlation between Alexis values and Lumipulse values for the same samples, you see there's a high correlation. So the Pearson correlations are denoted by an R, and the Spearman correlations are denoted by a rho. So they're high correlations, but they're definitely not 0.98. And interestingly, the Spearman correlations are higher than the Pearson correlations, suggesting that for some analytes, especially in this case, total tau, that the relationships between the Alexis and Lumipulse values are not linear. So the Spearman is a measure of rank correlation, and Pearson is a measure of linear. So when the Spearman is a good bit better than the Pearson, that tells you that the correlation isn't really linear. And also you can see that the absolute values for the assays are totally different. So this dashed line is the identity line. So if you got the same values on both assays, it'd be on this line, but they're not. So the Alexis values for A-beta 42 are higher than Lumipulse. For tau and p-tau, the Alexis values are lower. So how do we go about putting these values on the same scale? So certified reference standards have often been cited as a way to put values from different assays on the same scale. And you can actually go and buy low, medium, and high certified reference standards for CSF A-beta 42, but they're not being routinely used by most labs. Also, I would note that if you have 3 points and you have a line, that the 3 points should do a pretty good job of correcting for the differences. But when you have more complex relationships between the two assay values, if you have a low, a medium, and a high, that's really not going to do a good job of converting values depending on how you set up your analyses. So how do you deal with that? So in general, we've found that natural log transformation really helps to improve the model fit of fluid biomarker data. And that really may facilitate rescaling of biomarker values. Another thing that we found is running a group of samples, not 3 certified reference standards, but like 30 samples from 2 sites together can allow you to evaluate the relationships of the values at the 2 different sites. So basically you can come up with an equation for the value at one center versus the value at another center, and then apply that to rescale the values so that they're similar. Now, of course, there's caveats to this, and there's error in the estimate that will reflect differences between the 2 sites and the assays. And there's also, there could be systematic error related to pre-analytical factors that may be difficult to measure and account for. So in conclusion, I would say that harmonizing this data, even across 2 high-performance assay platforms, is actually pretty challenging. I would say that if we hope to do this, that reducing the number of assay platforms and also the number of lots is really essential. I think that bridging samples may be a way to harmonize biomarker values across sites, that is running a subset of samples from each site together. And the ideal situation would be to run all the samples together in a central lab on a single platform. And maybe Tatiana will discuss ways to do that. I would note that for some studies that may be reasonable, for other studies that may not be a reasonable idea. Finally, even after we carefully bridge these biomarker values together, it's going to be important to evaluate for consistent associations between the levels and the variables across sites. And this is particularly important because there may be site differences that we haven't measured or that might be extremely difficult to account for. Like a lot of these pre-analytical conditions may be hard to really adjust for. So in conclusion, I think that if we can combine our collective resources, we could potentially create more powerful data sets that improve the interpretation of biomarker values. And I think this has a potential to really improve the accuracy of identifying individuals with Alzheimer's disease. And happy to take any questions, or we can have Tatiana and Jeff Dage talk next.
All right, I think I'm up, so yeah, I think you can see my screen. I'm gonna put it into full screen mode here. So you can all see that okay? So yeah, thanks for letting us come and give you an update. So I think we provided some information a few months back on setting up a laboratory. Really jumps off of what Suzanne was just saying about providing reliable and consistent research biomarker results, where we can then compare through time and across studies, really to create some of these larger sample sets and things like that. And so our progress so far, so our immediate goals were to establish a laboratory and implement a few biomarker assays in a really reliable and consistent way. And they're highlighted there, and we're focusing initially on phospho-tau, neurofilament light chain, A-beta 1 to 40 and A-beta 1 to 42. Those will be the first 4 assays that will be in production, basically. This was our project plan we put forward before, and this is just highlighted again just to show that we're kind of on, we are not kind of on schedule, we are on schedule, and things are moving along really well. We purchased all the necessary equipment. We have obviously the basic lab equipment, but we purchased the TCAN Fluent system in order to automate all of the sample handling and have consistent dilutions and sample aliquoting and things like that, as well as to fully automate the A-beta 1 to 40 and 1 to 42 ELISAs. We have the Quanterix Simoa, which we're going to be implementing neurofilament light chain and the phospho-tau 181 kit assays. And so that's all been installed and is currently undergoing qualification runs. And I will point out that Eli Lilly did donate to us or transfer to us 11 neurofilament light kits to facilitate our training and things. So that was really helpful for that, just to kind of get things moving. So the priorities of the NCRAD lab, really it's, first and foremost, of course, is not just because it's the audience, but the ADRCs, and what we can do to facilitate biomarker sample analysis. And so within the grant for NCRAD, we have submitted administrative supplements. One of them includes the analysis of 4,000 ADRC plasma samples across a few years, 2022 and 2023, the next 2 years. And so we really do want to, through that supplement, we want to design the right assays. We want to generate, a lot of data that'll enable us not only to answer important questions about the disease and things like that, but to create these larger sample sets in a consistent way that can then be combined for even greater detailed analysis. So that will be the focus over the next couple of years for sure, or to try and work with all of you to kind of design the right studies, get the right sample sets, and conduct those analyses. And so we do want to encourage everybody who's thinking about doing that and maybe wants to use the NCRAD laboratory to do that, just to reach out. We can talk through the protocols and around plasma collection or processing, or what volumes we need, or whatever other details we need to kind of talk through. So just to kind of highlight, there's been a few people have already contacted us, and we've been planning different studies. And so this is just kind of highlighting kind of a snapshot, if you will, of proposed studies that we're contemplating, or some that are planned. And so you can just kind of see over 2022 and 2023, we have quite a bit at least being discussed and planned. And so this doesn't include a lot of ADRC samples. And so as you reach out, we do have a lot more capacity. Our initial target was 10,000 samples a year. And so we have plenty of capacity left. So nothing to worry about, but we are tracking it and we'll keep track of it. So, and really this is the key here is, from the NCRAD biomarker laboratory perspective, new grant submissions, we're encouraging banking with NCRAD, of course, to share biological resources across the research community, but also we can provide, consistency around protocols, manuals, training. If we do the sample QC and distribution, we can kind of, have a very uniform process for all of those steps. So again, new grants, if you're submitting for that, we can include funds to support the plasma biomarker analysis through NCRAD biomarker lab, and it can be facilitated and streamlined in the single by the single group that's handling the other banking requests. And then we are, as Suzanne pointed out, we are looking at several comparability studies across labs and setting up those studies, kind of exactly how Suzanne laid it out. And so if you're interested and you have your own biomarker lab set up and you're running some of these assays already, and you want to kind of bridge between laboratories, we are in the process of kind of planning those and designing how many we're going to do and collecting or obtaining the right sample sets in order to do it well. And so if you're interested, reach out for that as well. And we can just, include you in on those discussions and plans. And really that is about bridging to historical data as well as future data. So everything's on the table. And with that, that's it. So I'm happy to take any questions or have additional discussion.
You've got at least two. I'm going to let you do them, Jeff. So there's one from Suzanne Kraft. Can you please discuss considerations for analysis of longitudinal samples, i.e. is batching of baseline and follow-up samples needed? Do you want to start with that one? And then I'm going to add.
Yeah, sure. It's always best practice. So if you're looking at trying to look for really small effects, it's best to batch analyze and even run the samples together on the same plate. That'll minimize the variation that you see due to analysis effects. We are implementing them kind of, our goal is to have longitudinal stability of our assay and our implementation. And so, do you have to do it? No, you don't. The amount of variation you see within a plate is going to be smaller than what you see across months and months and years, such that like within a plate, it's going to be on the order of 5 to 7%, variation or error. But across longitudinal, across a year, our goal is still to target less than 10%. So it's not a huge difference. But it's, if you're saying, oh, I have all the samples, should I run them together? Yeah, you should, and you should run them on the same plate. If you're designing a study and you want to separate baseline from longitudinal measurements, we can do that too. It'll just be slightly increased error, but it's not going to be a dramatic difference because we're going to keep everything under control the way we're implementing our bridging procedures and bridging lots and instruments and operators and things.
Got a question, Tony. You want to just say it, Tony?
Yeah, thank you for the overview. I was wondering if there are any efforts to measure large-scale unbiased proteome markers. And the reason why I think that could be useful is that we find a lot of proteins are highly correlated with each other. And if we are able now to measure, up to 10,000 proteins reliably, we might find proteins that are very strongly correlated with other plasma biomarkers that we currently measure with very sophisticated, complicated tools. That might give us the possibility to avoid batch effects, or there may be markers that are much more stable, that are not affected by exactly how you spin down your sample, how you store it. And I feel we haven't really explored this as a field.
Yeah, and I think your presentation earlier kind of illustrated that really nicely when you measured it with the SomaScan. And I think we're not planning on doing that within the NICRAD laboratory, but it's certainly something that's a worthwhile exercise to measure those in a reliable way and in a consistent way. Like O-Link or SomaScan, something that's very reproducible and can be done at a large scale. And that's really what Michelle pointed out on p-tau. That's why that's a good predictor of A-beta pathology, right? Because A-beta measurements are hard. But that's the p-tau is a lot more robust and easier. So I think it goes to what you just said as well.
I don't know if you wanted to mention one other thing, Jeff, and I'm just looking to see if there's any other questions. So one of the things that came up at the last biomarker working group meeting was the question about investigators that might want to use these results in terms of as part of a screening to identify which subjects they want to enroll. So you've just made a good case for why you want to put samples together and run all your study together. I wonder if you just wanted to speak to that. I think some people have heard it, but I just want to make sure we cover.
Yeah, no, thanks. And I forgot to mention that. I said I was going to. So like I said, we are trying to have things consistent over time, and we should be able to do that. Still, for research purposes, that's what we're running in the lab. We are working with C2N to establish kind of a collaborative agreement where we can process samples through them as well, and that'll enable people who want to have samples collected in a CLIA type of laboratory. We can facilitate that. And so that'll be for plasma A-beta measurements. For p-tau measurements, if you want to use it kind of as a screening, as long as we're not needing to do CLIA level for return of results, we should be able to have consistent and stable analyses over time. And again, that's what our goal is. And so you should be able to set criteria and then stick to that criteria over time if you wanted to use it in that way. But again, it's research purposes out of our laboratory.
Hey, Jeff, you've got a question from Ron Peterson, wants to know about your plans for p-tau 217. You knew that.
So I'm currently still working with Eli Lilly to work on a material transfer agreement. Things are moving forward. It just takes a long time to set up those agreements, and our plan is to still try and put that in place so that we have the 217 assay as well. We chose the Quanterix 181 assay for implementation in the lab because it's commercially available. And as a kit-based assay, it's going to be widely available by a lot of people. So it seemed like a very attractive assay to move forward with. But the field is rapidly changing. So there's new platforms, there's new assays, from other companies as well. So I think the p-tau field as it moves forward is going to be a challenge for the NICRAD laboratory. And so we're initially implementing the Quanterix 181 assay. We have plans to bring online with Eli Lilly's help, the 217 Eli Lilly assay, but we'll watch the field. And as, again, Suzanne pointed out, if there's some of these fully automated platforms, Roche Alexis, Fuji Rabio, if they come out with one of these assays and demonstrate that it has better performance long-term and more stable, there's nothing that'll prevent the lab from switching to some of these other modalities or platforms, but it'll be something that we don't do willy-nilly. It'll be thoughtful and done in a way that'll be consistent with the best path forward. But thanks, Ron, for that. We are planning to do the p-tau 217 assay on the MSD platform with Eli Lilly's help.
I don't see anything else. Anyone else have questions?
I have a question for Suzanne.
There's also a question from Doug, but go ahead. We'll we'll let you ask Suzanne, and then we'll leave out Doug's too.
Yeah, it was a great talk. And when you were comparing the Fuji Rabio and the Alexis, it was really nice to see the A beta correlation, and then could be corrected, but then the non-linearity of the 181 and 2 total tau. And I wondered, do you think that's because they're actually not the same assay? Because I think you brought that up. Sometimes they measure different forms, and those two platforms, I think for total tau and p-tau, measure different forms of those analytes. Do you think that's the reason for that nonlinearity?
I'm not sure. I would say that just looking at the distribution of biomarkers, that across platforms, that the tau and p-tau and NFL is actually even worse, tend to be very log scaled. And so I think in nature, most things don't follow counting numbers, they follow logs. So I don't know. So it's possible, but it's also possible that it could just reflect natural patterns, just the way the tau and p-tau are distributed. Well, one other thing that I found interesting, and I didn't include it in this because we don't have a beta 40 data for Alexis, but interestingly, the distribution of the ratio A beta 42 to 40 seems to be a lot more normal, or actually it's bimodal, and that's not at all what we see with tau and p-tau. But with respect to bridging, I think it shows that we have to figure out how to bridge the data together, taking into account that it may not follow a nice straight line.
There was one last question, if we're allowed to do this one. So Doug asked about measuring some routine laboratory parameters centrally, and specifically calls out creatinine and CRP, to name a few. I think there's been discussion also from the NAC steering committee, potentially, or maybe it's just I've heard the chatter. There's certainly been discussion around doing that. So I don't think there's anything that would preclude that, but I think there would have to be an interest more broadly to do that, I would say.
Because again, I mean, the reason I bring it up is, we can all do this at our own
uniformity
hospitals and clear labs and whatever, but the assays may be different, the cutoffs may be different, and it may be tricky to integrate that sort of data.
Yeah, I agree with you, Doug. I think you cut up. Yeah. Oh, there you came back.
So, I mean, if we think of a few of these that are measuring biologically important pathways, maybe a centralized effort to get them measured would be highly productive.
was going to read one last comment. Or do you want to say something about that, Jeff?
No, I just agree with you, Doug. I think, for things like that we can show have a large influence, we should try and centralize it because it'll be more consistent that way if we can.
It would be. All right, there's still more. I'm so sorry. So can I assume Cerise will cut me off? There's two more questions that just came in. One from John Shao. Lots of centers have platforms and investigators able to carry out research quality immunoassays and are planning homebrew plasma ATN assays. Thoughts on how the ADRC program should deal with this? That's probably kind of a broader question for this broader group. I think we've said, at least as far as NICRAD, we're looking for commercial standard assays. That's sort of the approach we're taking because we think that's going to be the most useful thing for the field. I think it'd be interesting to hear what other people think and want to speak to ways to collaborate or potentially compare data. That's where I think some bridging samples would be very helpful.
Yeah. And I'll just say that, homebrew isn't a bad thing. I wouldn't choose it unless there's a reason. And so presumably people who are implementing homebrews, it's because they have designed the assay to be better and show something really unique, which I think is always interesting science and useful.
Still one more. I'm trying to make sure I get them all. We need an LBD measure. Sterling says RT-QuIC maybe in CSF, since there's often co-occurring pathology at autopsy. Are there any recommendations or central plans around that? And I don't know if anyone wants to speak to that. I mean, we don't. But
so sorry. I can comment a little since I've been involved in a number of groups and projects on RT-QuIC. If you do want to do anything centrally, there's really only one lab that has set themselves up in a clear-like way to do this, and that is Amprion. There are a number of academic labs that have set up RT-QuIC labs. I think in terms of reproducibility over months and years, and is the lab going to be able to do things in 2021 and 2022 kind of the same way? I mean, again, if there is interest, I would suggest that, again, people think about working with a group like Amprion. And again, this could be done centrally or through individual centers.
So the one thing I guess I'll add is one of the things we've been trying to do at NICRAD, and I'm not solving the RT-QuIC problem, but I guess I just want to put out the approach that we're trying to do. So we've been trying to see if there's ways that we can help make it easier if there are multiple groups that want to work with another laboratory to try to help with paperwork. So Jeff alluded to that in terms of A2N. C2N, if there were also questions around, for example, wanting to work with Amprion, if there was a way that we can centrally, we have CSF samples and can help coordinate that, we're really glad to do that if that helps institutions. I think it's something that if we've got the CSF samples, it's really something we're glad to help do. There's one other question, I don't know who it's from, that asks about whether or not labs should be turning to Lilly or Janssen to set up their own agreements in terms of being able to set up P217. I don't know that we're necessarily in a position to answer that. I don't know if you want to comment on that at all. I mean, I don't know if anyone wants to comment on that.
Yeah, I'll just make a general comment. Anybody's always welcome to reach out to anybody. In my experience with pharmaceutical industry partners is a challenge to set up agreements. So, if we can align and focus and have fewer agreements, I think the field will be in a better place as long as those agreements are structured to enable more people. And really that's the goal of what we're going to try and set up so that it's not going to be as restrictive as a single use agreement.
Okay. We also got a question about handling skin, urine, or fecal or stool tissue. So I think that one's for me. So I'm glad to address that. And Jeff, if you want to answer anything. So we can certainly, so across those of you who know, I mean, we run NICRAD, we also run an NINDS biorepository and also work with PPMI. So we are receiving currently stool as part of the American Gut Microbiome Project. So certainly are getting stool and have protocols around that. For urine, we actually have a lot more urine collections that occur in our PD studies. And so do have protocols for that and can receive that. And then skin also are involved in that in terms of PPMI. So in one of the That's the Fox biorepository. So do have protocols around that, if that helps. Oh, and then same question again, is the commercial P217 tau, is that going to be on MSD or Quanterix or both? Do you know about that, Jeff?
Yeah, I don't know that there's any commercial product yet, but we'll watch the field. I don't know that there is one. We're going to be implementing initially the non-commercial version, unfortunately, and that's on the MSD platform. If something becomes commercial in the meantime, we can certainly switch to that. But right now there is no commercial 217 assay that I'm aware of for plasma.
Anyone else? Any questions for also Suzanne? Glad to be your moderator for that as well. Anyone have any more questions? Okay. All right. Back to you, Cerise. Thanks so much. I think you might be muted, Cerise, if you're talking.
Hello, I was talking all that time. Can you hear me?
We can hear you now.
All right. So I want to thank Suzanne and Tatiana and Jeff for talking about logistical challenges and updates. I put this at the end of the meeting because it's, highly important that, we move on to the next steps collectively and think of ways to overcome the challenges. And Grayson is sharing my slides again because I'm going to try to go through my presentation on the phone and over the next 5 minutes. And because I have some discussion questions to lead into a discussion period. So basically at today's meeting, we gave you like an overview of many things that are happening in the field. And I want all of the information that is on this timeline to serve as a baseline for all the volunteers who will serve on the Biomarker Steering Committee coming up next year. Next slide. So hit it again. And we started thinking collectively about what the center can leverage by gathering all the data that you've provided prior to 2020 in table A7, and we summed all the biospecimens that are available across the centers. And in 2020, we discussed having these biospecimens matched in NAC and NAC coordinating to have these biospecimens matched to the clinical data, and so that at least we would have that database kind of taken care of. And that's still something that needs to be considered, and these inventories probably would need to be addressed again. Next slide. We revamped table A7 for the last reporting period, and we had a long survey for you to fill out. And we sectioned it by all the different kind of biomarkers based off the past table and then the new iteration. So we have a neuroimaging section where we are asking you in table A7 if your center has a neuroimaging core in addition to the required biomarker core, and yes or no, you collect amyloid PET, tau PET, or other PET and MRI. So these are yes/no questions. And then if you do amyloid PET, list the ligands that you use. Tau PET, what are the ligands that you use? If you're doing other PET, list what the other PET targets are. And next slide. We kind of we have it in the summary that, all the centers at the data collection collects MRI, majority collects PET, and half of the centers do some other PET ligands. Next slide. The next kind of chunk of the survey is about participant blood and stool. Again, you use this table for a yes, no. You collect cell pellet, DNA, RNA, plasma, serum, white blood cells. You make lymphoblast cell lines, you make fibroblast cell lines, or you collect stool. So a yes, no. And next slide. From what we got in the last progress report period, the majority of the centers collect DNA, plasma, serum, PBMCs, and whole blood, and less of the other components. Next slide. The next section of the survey is about blood analysis. Again, you answer these by yes, no. You conduct blood amyloid beta 42 or total tau or phosphorylated tau or ApoE. And the results last year were, the next slide, that majority of the centers are doing ApoE, but we're still 50-50 on having all the centers quantitating the blood analytes here. Next slide. CSF, again, when you answer this, it's yes, no. You conduct a test to quantitate in the CSF these 4 markers. And the results from last year were that majority of us do collect CSF and we do quantitate amyloid beta 40, 42, total tau, and phosphorylated tau. Next slide. And then we kind of wanted to know about your plasma analysis tools. Are you using ELISA for quantitations, and what targets are you using ELISA for? Are you using Quanterix? What targets are you measuring? Are you using Roche? What targets are you measuring? Are you using mass spec? What targets are you measuring by that? Then also there's like a other biomarkers section. Are there, is there anything that wasn't accounted for in the above categories? And this will change by center. Then there's like a little extra note section. If you have any special conclusions or considerations about the biomarker core that's not accounted for in the table. And in the next slide, the result was still kind of 50-50, that half of us kind of use these methods for quantitation. And so I think that brings us to the next slide, where we have these questions for discussion. How did the inventory or that checklist improve your understanding of the biomarker landscape of the center network? What immediate data collections can we maximize across the network to contribute to the understanding of the disease? And how can we prepare biomarker catalogs and upload the relevant data to NAC? So I think for the last year or so, these are kind of our meaning NIA kind of 3 main questions that we'd want the steering committee to begin with when they are established next year. So everyone at the meeting knows what those 3 questions are now. So, and I'm open for the next few minutes to have, you can raise your hands and we can have a discussion. We can have follow-up questions of the presenters. And I thank you all for attending this session.
Hey, Cerise, I raised my hand. Maybe since no one's talking, I'll just jump in. I think this is really helpful, and it's really helpful for me, at least, to see the different breakdown of what the centers are doing. And I will say, as far as the plasma biomarkers go, I think there is a lot of value, and especially when I see some of the data that Tony was sharing earlier. the the 4,000 different proteins and the multi-organ correlations. Having these individual measures, like especially phosphorylated tau, in that analysis to look for correlations and improve the correlations, I think it's going to be really helpful. And so, that's my 2 cents, is I'd encourage people and centers to, start regularly measuring the plasma biomarkers, because at least for the phosphorylated tau, they work really well, and they are showing you elements of Alzheimer's pathology, so it can be really useful.
Thanks, Jeff. Krista wrote in the chat about one additional dementia is what we collect versus what we're able to share for one reason or another. And I just wanted to say, thought, just like an administrator, I appreciate that. And that's very true. And when we, did the summation of the possible biomarkers, again, they're just kind of like numbers that are available, but, the devil are always in the details. And, there are many different reasons why, some biospecimens exist, and how many of that is translated into, sharing. Yep, it's definitely going to require a lot of work. I'm always open for any kind of activity. Maybe we just need real life to happen again one day.
Hey, Cerise, I was going to make one comment maybe on the last item, thinking about catalogs and uploading relevant data. And I just want to put it out there. And maybe I missed this, as we've been talking about, the importance of bridging and the importance of what other samples were run so that we can more effectively combine data generated in other laboratories. I don't have an answer, but I would think that would be an important aspect of the catalog, is the ability to know, do you understand what I'm saying? How you'd be able to put together data from different laboratories. It seems like you would need to have some information, again, not only on how the assay,
like what the specimen is, but how the specimen was prepared.
Yeah, well, I guess I'm just asking. So, again, thinking about, just variability in assays run at different times, run at different laboratories. So I don't have an answer for it, but it's something we probably need to think about.
Most definitely. Thanks, Trey, for your question. He says the earlier idea of centralized soma scans and O-link assays is appealing for prioritization. These data are especially valuable when done on large sample sets and are connected through clinical data through NAC. Thanks for that comment. Oh, I see Tim is in the audience, and Tim is, the PI of a large U24 harmonization project. Do you have any insights that you learned from the meeting today, or that some roadblocks that you might have witnessed that you didn't know about as you're working on your project?
Wasn't trying to get called out, Cerise, but I'll take it. No, it's great. I think the point that Tatiana just made about, the more we can document what was done and how it was done, the better off we're going to be on the harmonization side. But to Trey's point, ideally, if we run it at one center and one site like that, you take the problem out of my hands, and it now becomes an assay issue instead. And that's where we'd prefer to be. It's a lot easier to fix the problem on the front end than to try to do statistical tricks to fix on the back end like what we're doing. So.
Thanks. I appreciate that. Well, the information that I shared in my presentation is going to be uploaded to the calendar invite so that you all have it. Hannah is going to do that from NAC. This meeting has been recorded and will be available on the NAC YouTube channel. When it's available, Hannah will send out a message from NAC. Again, I thank you all for taking the time to attend another Zoom meeting. What else? Look out in January when NAC begins the election process, and I hope that you take today's meeting and our thoughts and our questions and the past meetings over the last 3 years of whether you volunteer to be part of the steering committee. So we're looking for 7 individuals. So if there isn't any last minute thoughts or objections to the whole thing, then we can dismiss. I appreciate your feedback. Thank you. Have a great afternoon.